Thanks everyone for coming to our exciting session today. We're going to be talking about constraining the dark ocean carbon cycle and hopefully talking about whether or not we think these processes are really important for the overall ocean carbon cycle in general. So I just wanted to start us off by giving a little bit of an overview for what we're going to be covering in this session. And so, as I mentioned, this session is about dark inorganic carbon fixation, and we're really excited to have this session because it's a process that's kind of poorly characterized overall. So in terms of what's poorly characterized, we don't really have a good handle on who the kind of the key players, the different rates, the particular organisms that are involved, and definitely how this relates more broadly to kind of the overall global carbon cycle. And that's kind of one of the major goals of OCB, bringing all of these folks together. What we do know is that budgets of the deep sea carbon cycle are currently unbalanced. And so one thought is that if we could better characterize dark inorganic carbon fixation, that we might be able to have a more complete understanding of the marine carbon cycle. And hopefully all of us will be able to discuss whether we're on the right path to that or what are some of the missing pieces to that today after this session. So we have a lot of exciting talks, and they're ranging from kind of discussing rates of inorganic carbon fixation in lots of different ocean habitats within what we consider the dark ocean, so just below the euphotic zone. We're going to hear talks that are using more omics-based approaches to looking at this and specific kind of metabolic pathways. And then we're going to also have kind of a broader perspective on carbon cycle modeling, for example. And one thing that I wanted to mention is that with our OCB summer workshop, we often use sessions like these as kind of a jumping off point for bigger activities. And we often also have sessions at OCB that are kind of on these really exciting and emerging topics or places where there's a lot of kind of knowledge gaps. And so just kind of keep that in mind as we go through the session today. This might be a topic that you're learning something new about that you haven't really heard a lot about. And maybe you've heard about it, but you haven't thought about how it fits within kind of our understanding of the global carbon cycle. And so at the end, when we move into kind of our more discussion part, hopefully that'll be a place where you can lean on your speakers, talk to them more, and try to understand how this fits into our broader understanding of the carbon cycle. And maybe the session might be a jumping off point for future OCB activities, et cetera. So I just want to give a quick overview of what the schedule for this session looks like. So we're going to have a series of longer talks and some lightning talks. First, we're going to hear from Dekas and many of her lab members, and they're going to give us kind of a primer on what we're even talking about with dark inorganic carbon fixation. What are the relevant processes? So this is going to be a really cool overview and roadmap for where we're going. And then we're going to hear from Alyson Santoro, and she's going to talk about some of her lab's work on nitrogen and nitrification in the mesopelagic. Then we'll hear from Sarah Hughes. She's going to be talking a lot about Protists and how does inorganic carbon kind of move up the food chain within the dark ocean. And then we're going to have a couple of lightning talks from Nicola Paul and Hope Irene. And then we will have a short kind of bio break. And then we're going to hear from Stephen Haddock. He's going to talk about metazoans and secondary consumers. And again, also this kind of like broader scale, how does this fit into global carbon cycle? And we'll finish up with a few more lightning talks from Saeed, Soon, Kayla, and Renjan. And then finally, we're going to have a panel discussion. We're going to do this a little differently than some of the other sessions discussions. We're going to have some breakout rooms, but I'll explain that more when we get to the end. And so with that, I can stop here and we can start moving into our session.
Perfect. Thank you, Randie, for that great introduction. I'm really happy to be here today, and I'm excited to give this kind of overview talk together with a bunch of my group members on what dark inorganic carbon fixation actually is and why we think it matters. So To start out, a little introduction to the deep sea. So the vast majority of marine research, of course, takes place in the surface waters, the euphotic zone, and this is both because samples are easier to collect there, and also because a lot of productivity happens there. Things are growing densely and very active. But the rest of the ocean is very important to understand too. It covers 2/3 of our planet. It's this enormous volume, and because of that, it's storing and transporting nutrients and other biological and chemical entities that are really important all across our planet. So it plays a really important role in the Earth system, yet it remains really understudied. So an example of this in the microbiology realm is that as you go down with depth, as you can see here, the average 16S gene sequence is more and more dissimilar to those that are in publicly available genomes, for instance, in NCBI. So there's a lot of biological novelty at depth and a lot of questions about how these organisms are participating in biogeochemical cycles and climate. And as I was hearing some of the talks this week, I was reminded of how many of the solutions we have to our climate challenges involve sinking organic matter into the deep sea, and how little we know about the rates of remineralization there, and how particularly important it is, therefore, to get a better grip on this. So a very simplified view of carbon cycling in the deep sea looks something like this, where we have carbon dioxide fixed by photosynthetic organisms at the surface into organic matter, and that eventually sinks down to the seafloor. Something like 1% of that organic carbon actually makes it to the bottom. Like I said, there's a lot of mysteries about what's happening in the dark ocean here. And one of the main reasons we know that we don't know it all is because there's a discrepancy, as Randie just referred to. So the sources of organic carbon to the deep sea that we know of are much smaller than the measured sinks for organic carbon. And this is essentially impossible. So it means that either our rates are wrong or our inventories are incomplete. And one of the things that I am interested in thinking about in this context is the potential for underestimated dark inorganic carbon fixation. So the potential for carbon to be fixed at depth and contribute to this cycle. So what is dark inorganic carbon fixation is the question that we're going to be talking about in this presentation. It is a biological process in which inorganic carbon, so that's bicarbonate or carbon dioxide, is assimilated into biomass without using energy from light. So an autotroph is one of the major players in this process. So this is an organism that can generate the majority of its biomass from inorganic carbon. Most of you are probably familiar with that idea. Autotrophs are broken up into photoautotrophs and chemoautotrophes, and today we're going to be focusing on chemoautotrophes because these are the ones that are functional at depth. Heterotrophs can also fix inorganic carbon. So they do this when they are performing their regular anabolic processes to create biomass. And so they need to be taken into account in this discussion as well. And the more we learn about organisms, particularly in the deep sea, we see that it's not just a binary split between autotrophs and heterotrophs. There seems to be a lot of potential for mixotrophy and metabolic flexibility. So we need to think about this microbial lifestyle as well. We'll be talking about all three of these different categories. Rebecca Salcedo will start first with chemoautotrophy, Alex Jaffe second with mixotrophy, and Emily Parrish third with heterotrophy. These are all members of my group. And fourth, we'll have Chloé Baumas talking about so what? So how does this really relate to that carbon discrepancy that I was mentioning at the beginning? So before I turn it over to them, I just want to mention a few of the methods that we use to study dark inorganic carbon fixation. So we use DNA and RNA analyses, so these are metagenomics and metatranscriptomics, to see the genetic potential of the organisms in the environment. The genes involved in chemoautotrophy, for instance, are very different from those from the heterotrophic inorganic carbon fixation. And we can also use bulk uptake of radio and stable isotopes to see the full rates of inorganic carbon fixation. And this is very useful to get a handle on actually how big of a contribution these processes are having on the global cycle. We need, though, kind of more specific techniques, and particularly single cell techniques, to be able to differentiate that bulk inorganic carbon fixation signal to attribute it to either autotrophs or heterotrophs specifically. So the way that works is that we'll do a stable isotope incubation with both the 13C labeled, for instance, bicarbonate, but also 15N labeled ammonium. So in this case, the 13C bicarbonate uptake is telling us how much inorganic carbon fixation is occurring. And the uptake of 15N is we're using as a proxy for total growth. So using the nanosims, we get an isotopic composition of each individual cell, and then we can look at the ratio that they take up, both from bicarbonate and ammonium, to separate them into their metabolic lifestyle, so autotrophy and heterotrophy. So for example, here you can see for a given amount of inorganic carbon uptake, you could have a cell that makes up a very large percentage of its biomass, so it comes across as an autotroph, or a small percentage of its biomass, and then it is actually a heterotroph. So looking at this on the single cell level allows us to differentiate this signal into the different groups and therefore mechanisms responsible for it. All right, with that, I will hand it over to Rebecca.
Hi, everybody. I'm Rebecca. I'm a PhD student working with Anne, and I'm going to keep talking about chemoautotrophy in the deep sea. So as Anne mentioned, chemoautotrophy is a metabolism where cells can power inorganic carbon fixation using the oxidation of chemical compounds. And this is a metabolism we're finding all over our planet across a diversity of habitats, both in the marine realm as well as the terrestrial realm. There are 7 different pathways. There are 7 known pathways that organisms can use to perform inorganic carbon fixation. And these pathways vary in their energetic requirements as well as their phylogenetic distribution and biochemistry. And they also have key genes that we can use to look for to assess chemoautotropic potential in things like metagenomes. Chemoautotrophs harness a diversity of chemical energy sources. Really, you just need any reduced inorganic compound. So here is a very small, non-exhaustive list of potential energy sources. This is something that is continually growing as omic and culturing efforts continue to expand it. When we start looking a little bit more specifically into the marine chemoautotrophs and the pelagic chemoautotrophs, I kind of have to mention AOA. They are probably our most abundant as well as our most well-known pelagic chemoautotrophs. So in certain areas of the ocean, such as the Hawaiian time series, AOA can reach up to 40% of the cells in the dark ocean. So these are quite important players. This is something our following speaker, Alyson Santoro, is going to talk much more about. However, ammonia oxidizing archaea are not our only pelagic chemoautotrophs. So in addition to them, we also have nitrite oxidizing bacteria. These organisms complete nitrification together with AOA. There is some kind of continually rising evidence that the oxidation of diverse sulfur species is starting to become implicated in chemoautotrophy. We're seeing this a lot in DNA and RNA evidence, but also some incubation-based studies. We're also seeing the rise in the possibility for trace gases to support chemoautotrophy. So this includes compounds like carbon monoxide, hydrogen gas, and methane. And then in some areas of the ocean, such as oxygen minimum zones or oxygen deficient zones, we see that the anaerobic oxidation of ammonia, or anomox, is also important in relation to chemoautotrophy. But there remains this question of kind of what is the distribution of all of those organisms and these metabolisms across the water column, and how abundant are they? So we started to try to assess this question. We sequenced 28 metagenomes off the coast of Northern California and looked for the genetic potential of chemoautotrophy in the microbial community sequence. We see that these organisms are actually very abundant. So 150 meters and below, cells that encode the potential for carbon fixation account for 20% of all of the deep sea cells. They also encode 4 of the 7 carbon fixation pathways. This includes the Calvin cycle, which is something Alex is going to talk more about briefly, or more about soon. Oh, okay, there it is. They're also very ubiquitous. We detected potential for chemoautotrophy in all of our samples, including the surface ocean. We see phylogenetic diversity, so we recovered 17 different phyla that have this potential. And we also see that in addition to being phylogenetically diverse, they are catabolically diverse, meaning that we recovered genes for ammonia oxidation, methane oxidation, carbon monoxide, and sulfur oxidation across this community. Something that's interesting, and again, something Alex will pick up more on following this, is that we also observed some metabolic flexibility. So genomes that encoded inorganic carbon fixation pathways also contained more than one catabolic pathway. So we're observing some very interesting, at least genetic potential within these genomes. Of course, there's always knowledge gaps. So I think that there is a huge wealth of knowledge coming out about the kind of genetic and metatranscriptomic potential of these organisms. But something that is still missing is the actual activity. Are they fixing carbon? If so, how much are they fixing? What sort of controls are exerted on that activity? So I think that's something that still needs to be addressed. In addition, I think as microbial ecologists, we're kind of always on the hunt for novelty. So things about novel phylogenies or lineages, fixing carbon, are they using novel energy sources, things like that. We actually just got back from a 2-week cruise. So hopefully all of these questions will be addressed with that data and our knowledge gap will continue to shrink. So with that, I'll pass it on to Alex.
Great. My name is Alex. I'm a postdoc in Anne's lab. I'm also helping organize the Bay Area OCB meetup this afternoon. So if you are in the area, you want to come down to the peninsula, and feel free to get in touch. We'd love to see you. So today I'm going to talk about some of the work that we've been doing in the area of mixotrophy and metabolic flexibility. So just to kind of review what we mean by this, I'm going to build on this picture of microbial metabolism that Rebecca set out for us so nicely. As she mentioned, we're really interested in organisms that are capable of this process of chemoautotrophy in the dark water column. So harnessing chemical energy to fix inorganic carbon into biomass. We have increasing evidence that some cells that perform this process are not only capable of autotrophy, but also heterotrophy. So in the same cell, they can also assimilate organic carbon into biomass together. So what we refer in this case to as mixotrophy is really this capability of cells to derive significant portions of their biomass, both from organic and inorganic sources, either simultaneously or over time. The evidence that's accumulating for mixotrophy in the deep sea comes in a couple different bins. Is gene co-occurrence. A number of studies, including some of the work that we've done, have looked at genome sequences, primarily of bacteria that have been derived from the dark ocean, and looked at the co-occurrence of genes or pathways involved in carbon fixation, as well as genes or pathways associated with the transport or metabolism of organic carbon sources, for example, ABC transporters. So when we see these in the same genome, that implies to us that this organism has the genetic potential to do both and potentially to act mixotrophically. We also have some evidence from single cell activity-based assays in our lab. So this is a kind of similar plot to what Anne showed you before, with ammonium uptake on the x-axis and on the y-axis an organic carbon uptake. And in addition to kind of these clearly resolved populations of cells that look like autotrophs, that look like heterotrophs, we see these kind of cells in the light purple here that fall somewhere in between, that derive significant portions of their carbon from both sources, suggestive to us of mixotrophy. And this is from some of Emily's work that she'll talk about again shortly. So despite these kind of accumulating sources of evidence, they primarily come from single lineages or a small number of lineages and a few sites. And what's really lacking is kind of work that addresses the significance of mixotrophy over larger phylogenetic or geographic scales. So this is something that we've been working on in our group. As many of you will know, there is an enormous genomic corpus available from the ones that we have used to study autotrophy and mixotrophy. I, in particular, have done some work to assemble a large database of genome sequences from organisms that encode the enzyme rubisco. This is the key enzyme of the Calvin cycle, which is one of those and the most abundant carbon fixation pathway that Rebecca mentioned earlier. Rubisco being that kind of key enzyme that fixes inorganic carbon and turns it into biomass. Once we have these set of genome sequences, we can use those to basically probe the metatranscriptomic data sets that have been collected from across the world's oceans and align metatranscriptomic reads to our genomes to get a sense for how organisms in a given place and a given time are actually expressing their genetic repertoire. So when we do this, we see that rubisco gene expression is actually highly variable and it's non-constitutive. And what I mean by that is that it's not always on. I'm going to show you just a quick piece of data. So here I've got rubisco expression, or the rubisco expression on the x-axis. On the y-axis, we have depth, and each point here represents a single organism in a given time, in a given place for which we were able to resolve one of these gene expression profiles. And what you can see is that there is a wide distribution of expression values for this core carbon fixation gene over time and space. If we look at that for organisms that we think of as obligate autotrophs like cyanobacteria, or more or less obligate, rubisco is always very high. That's what we expect. But for some of the non-cyanobacterial bacterial lineages that are likely to be metabolically flexible. In these ways, we see more of a uniform distribution. And what I'm not even showing you here are the zero values. So it actually turns out that when you do this analysis, more than half the time, about 50% of the time, 55% of the time, Rubisco is entirely off, despite the fact that the organism overall is active. So when we see this pattern, we infer that, okay, Rubisco is off, but the organism's active. They must be assimilating organic carbon to support their growth instead of doing this autotrophically. So this is just another way of kind of probing patterns of potential mixotrophy over large geographic scales. Of course, there's still a lot more to learn here. We'd really love to know what governs carbon utilization in these organisms, right? Really, what is it that dictates one strategy over another? We hypothesize, but it is not really well established that this might be linked to geochemical gradients, particularly that of organic or inorganic carbon availability. We're also really interested in the extent to which these processes are spatially or temporally separated, right? Are organisms actually capable of assimilating both organic or inorganic carbon simultaneously? Does this happen in a spatially or temporally separated fashion? We don't know, but we would like to know. And of course, right, what this is all leading, getting a sense for the biogeochemical impact. What I did not show you today are some data from the metagenomic side that really shows these metabolically flexible, potentially mixotrophic organisms are very widespread in the global ocean, particularly the dark ocean, and are very highly abundant, as Rebecca's data point to. And so really understanding what it is that leads them to perform one metabolic strategy over another will really help us constrain their biogeochemical and other contribution to the carbon budget longer term. With that, I will conclude and hand it off to Emily.
All right. Hi, everyone. My name is Emily, and today I'll be talking about an often overlooked aspect of the carbon cycle, but increasingly important aspect, and that's inorganic carbon fixation by heterotrophs. So all heterotrophs can fix inorganic carbon, including us, even though they're defined by their reliance on organic carbon. And this can happen through a variety of ways, and most frequently talked about is anaplerotic pathways. And this is where heterotrophs assimilate CO2 into biomass through regular metabolic processes. And they do this often with the enzyme PEP carboxylase. This fraction of biomass that comes from CO2 can be anywhere from 1 to 15%. And we know this from a variety of pure culture experiments. So this one that I'm showing here is the heterotrophic Bacillus subtilis culture, where they assimilated or they sourced 2% of their biomass from bicarbonate over a 2-day incubation period. And there's a growing body of research about how heterotrophic inorganic carbon fixation is important in the environment. So on the left here, we have a recent preprint showing high expression of genes associated with PEP carboxylase, which is associated with anaplerotic reactions in the mesopelagic ocean. And then on the right, we have a correlation between heterotrophic production and dark carbon fixation from another recent preprint and some of the authors are speaking later today as well. And so together, these authors estimated that heterotrophic ICFs contributes about 30 to 40% of the total CO2 fixation occurring in the deep sea. And to add to this research, I specialize in using dual-labeled stable isotope incubations and NanoSim's single-cell analysis to quantify the fraction of heterotrophic biomass coming from CO2 and environmental samples. So today, the example that I'll be sharing is from a massive deep-sea brine pool in the Gulf of Mexico called Orca Basin. And this basin is a recent candidate for carbon sequestration processes. So carbon cycling in this basin is of interest. And here in the basin, oxygen decreases while salinity increases, creating this highly stratified redox climb. So as Anne mentioned earlier, we can characterize cells by their metabolic mode based on the ratio of bicarbonate to ammonium uptake in individual cells. And we can see what the fraction of biomass sourced from inorganic carbon is in heterotrophs based on the slope of this line. So here in the oxic-anoxic interface, heterotrophs sourced about 6% of their biomass from CO2. And this fraction is variable with depth. We don't really know what drives the magnitude of biomass that comes from inorganic carbon yet, but we can use this to calculate the contributions, the relative contributions of autotrophs and heterotrophs to total inorganic carbon fix with depth. And so what we can see here is that when you integrate with depth, about a quarter of the total ICF is the responsibility of heterotrophs in the redox client, and almost all of the inorganic carbon fixation occurring in overlying seawater is due to heterotrophic inorganic carbon fixation. And so we have a lot of the single cell data in a variety of terrestrial and deep sea environments. And I think what's next to kind of uncover here is what's driving the magnitude and rate of inorganic carbon fixation by heterotrophs in these environments, and how significant is it to the global carbon cycle. So with that, I'll hand it off to Chloe to talk about significance.
So hi everyone, I'm Chloé Baumas, postdoc in the Dekas lab. So in this presentation, we discussed in detail dark CO2 fixation, but the big question is, does it matter? And so just as a reminder, the biological pump is defined as the sum of all bioprocesses that export carbon, mainly as sinking particles, to the deep ocean to be sequestrated. And this ecosystem service is extremely important as it considerably reduces the concentration of CO2 in the atmosphere. And obviously, not all the carbon exported reaches the bottom. Sorry. It is estimated that only 1% of the carbon containing material at the surface finally reaches the sediment, where it can be stored almost definitely. So the rest of this organic matter is respired and remineralized, mainly in the mesopelagic zone, and the CO2 will eventually return to the atmosphere. So the amount trapped may seem derisory, but what's essential is the duration of sequestration, which is geological timescale and not the quantity. And so in parallel, without photosynthesis in the dark ocean, particles are considered the main source of organic matter. So the quantity of carbon exported here in green by the particle will decrease with depth as it is used, fragmented, transformed by organisms. And so the comparison between carbon export by particles and the demand for carbon by the organisms should tell us what is not used and therefore continues to sink. However, all the processes involved are difficult to quantify today. And one illustration of our difficulties is this discrepancy between carbon demand and quantity of carbon provided. So by focusing only on prokaryotes, studies show a difference of a factor 2 to 4 between the two, which is just impossible. You cannot have more carbon used than carbon available. So this discrepancy is known for decades now, and some possible reasons for that are, for instance, on the carbon demand side, the use of poorly constrained parameters, as illustrated by this plot, showing the exact same data with different values of conversion factor or prokaryotic growth efficiency. Or, for instance, the fact that 87% of the water column is under significant hydrostatic pressure, and mainly for technical reason, we keep analyzing prokaryotes' activities on decompressed sample. And on the carbon input side, there could be many reasons. Most of them actually have been discussed on Tuesday in a previous session. And in parallel, in the JETSON community, we are currently working to create a baseline of the biological carbon pump divided in 5 working groups, including working groups that are dedicated to carbon inputs. And what I noticed is the dark CO2 fixation, that is an in situ production of organic matter, is almost not mentioned in those efforts. However, previous work, shows some evidence that this could fill some gaps in our carbon budget. So for instance, in the North Atlantic, dark CO2 fixation and microbial heterotrophic production are of comparable magnitude. And in the Dekas lab, we published a paper last year showing that ammonia and urea oxidations could account for 5 to 7% of the POC fluxes entering the mesopelagic zone, which is not insignificant. And so to go further, my current work is focusing on reassessing those numbers, accounting for in situ hydrostatic pressure. And my results here are still preliminary, but using nanosim single cell technique that Anne introduced earlier, we observed that dark CO2 fixation here is higher under pressure in the dark ocean, which could decrease even more the discrepancy than previously thought by increasing this carbon input source here. And so to answer the big question, could dark inorganic carbon fixation fill some of the budget gap? Well, likely yes, because dark ocean carbon fixation could help, as we see, increase the carbon input part. And so the problem is data remain too scarce to constrain climate models without relying heavily on assumptions. So for instance, big models like PSCES or SAMS, among others, can include chemoautotrophes module, but it's not systematic and often overlooked. So our work in our lab aims to provide the necessary measurements to better understand and integrate this process. So there is as well a big effort to do in terms of linking metagenomic data or omic data to rates, because we have now more evidence, as you saw with the previous talk, with a pathway under omics data. And so as a long-term goal, this really matters for improving future climate predictions and informing like science-based policy decision. So yeah, with that, I can let Anne finish.
Perfect. Thank you, Chloe, Emily, Rebecca, and Alex. I think we would like to thank our other group members for contributing to the development of these ideas and data, current and past members, as well as some of our collaborators from around the world who have contributed. And finally, our funding sources, both internally from Stanford, NSF, NASA, and the Simons Foundation. And we thank all of you for your attention. And I'll just leave this kind of on the final point. All of these different types of inorganic carbon fixation, so whether that's chemoautotrophy, inorganic carbon fixation by heterotrophs, organisms that are doing a combination, are all contributing to this in situ production of organic carbon in the deep sea. And I think it's important to better constrain it. Okay, thank you.
Thank you so much. I'm sure everyone else agrees, but I already learned so much just from that introductory talk. We have a couple minutes if anyone has questions for either Anne, Rebecca, Alex, Emily, or Chloe, or just for the broader group, and then we'll switch over to our next speaker. Yes, go ahead.
Hi, everyone. Thank you for the very interesting talk. Do you have any clue about the role of dark inorganic carbon fixation in the photic layers, how they interplay with photosynthesis, that kind of things?
Yeah, that's a great question. So it definitely does occur in the photic zone. We measure active chemoautotrophs in the photic zone. And similarly, all the heterotrophic activity in the photic zone is also contributing through the same processes that Emily mentioned. In terms of quantitative measurements or like timing, I don't know off the top of my head, but that would be interesting to think about. When we were thinking about putting together this session and the idea of dark carbon cycling, we specifically didn't make it just deep sea carbon cycling for exactly the point you're bringing up. Even the surface ocean is dark half the time. So certainly these processes are important there too.
Yeah, hey, terrific tag team effort, you guys. What about light in the dark ocean? Are there enough photons to generate a bit of this carbon that we're also interested in?
Yeah, that's an interesting question that I don't know the answer to. Does anybody else on video know? Something that I think that's worth looking more into. I mean, off the top of my head, I would guess that there's not a ton, but maybe.
Maybe we should just sit around and eat some cake and talk about it sometime.
Sounds great. Good topic for breakout rooms later.
Right, still.
There's some questions in the chat. One from Yuki. Have you looked at dark carbon fixing microbes in aggregates or marine snow?
Chloe, do you want to comment on that?
Sure. I love marine stuff. We in our group, no, not directly, but there have been some paper that show some evidence, like specifically in micro niches in the particles. Some like particles are very like not permeable, and so you have like micro anoxic niches, and so that have been shown that you have chemoautotrophy there. But I have samples that I'm planning to analyze under nanosim for that later, so I'm very interested in those questions too. There are not a lot of paper working on that yet. Yeah.
Great. There was another question that I think Maria and Carly had, unless, sorry, Anne, if you had one more thing to add to that.
No, that's just what I was gonna to call
Oh,
out,
yeah.
that
Okay,
question.
yeah.
So the
The
question
question
is,
is just, yeah, go ahead, go ahead.
do you have any hypothesis as why high pressure might increase dark fixation? And so, yes, people have thought there's different schools of thought on this, but for many years, there's been a hypothesis that in general, rates of microbial activity measured in deep sea samples once depressurized underestimate the rates of true activity because these organisms are likely adapted to their in situ environment. So when we depressurize them, their rates would be lower. The idea then is if they were measured under pressure, the rates would be higher, which is what we did observe as Chloe showed. However, the situation is a little bit more complicated than that. In some studies that look at the effect of pressure, you actually see an increase of activity in some groups or in some types of activity. And that's thought to be mainly attributed to organisms that aren't always evolved and adapted to the deep sea, but instead have more recently come down on marine snow or have been transported more recently. And those organisms might become more active when depressurized because that's actually the pressure that they observe. So there's an ongoing discussion in this field right now about which of those kind of two processes are the overlying or like the most important one, whether it would be more or less active when depressurized. But I think it makes sense that it would be specific to the type of activity and the group, and maybe even different locations where you have different amounts of input from the surface. So complicated situation with multiple factors at play.
Great. Thank you all so much. I think there's a couple other questions going on in the chat that you can feel free to answer as we move on to Alyson.
Thank you so much for the invitation. I see some of my very best friends in oceanography on the call, and I'm really sad that we can't be together. So that was such a great setup, and thank you so much to Anne and her group for featuring some of our work, and I'm going to get a little more into that. So nitrification is what I'm going to talk about today, and if you don't remember what that is, it's the microbial oxidation of ammonia, or NH3, to nitrite, which is NO2, and then a second group of organisms catalyzes the oxidation of nitrite to nitrate, NO3. And this process links the most reduced and most oxidized forms of nitrogen, and so plays a pretty obvious role in the marine nitrogen cycle. But what I want to talk about today is how nitrification as a process interacts with other elemental cycles that we might be interested in. So these are aerobic processes, and so they impact the oxygen cycle in the ocean. 2 moles of oxygen are required to oxidize a mole of nitrogen, not quite in this stoichiometry here. These organisms also use a set of very interesting enzymes to catalyze their reactions. Metalloenzymes that use iron and copper to carry out their oxidation reactions and carry electrons in their shell. So because of that, we think they probably have some really important impacts on trace metal cycles that we're only just starting to understand. But the focus of our talk and our time together today is to talk about how these organisms, as chemoautotrophes, fixing carbon, impact the carbon cycle in the deep ocean. And I think it's important to remember that we're not just talking about arrows on a biogeochemical diagram, we're talking about reactions that are catalyzed by cells. And therefore, the ecology of these organisms and their physiology means something when we're trying to understand how these arrows work. And so this is a picture of some ammonia oxidizing archaea that I cultured during my postdoc, and they're really tiny. These holes on this filter are about 0.2 microns. So these cells have the ability to fit through some of these holes, which is pretty amazing. Second step of nitrification is carried out by nitrite oxidizing bacteria. These are some amazing electron micrographs from Stan Watson and John Waterbury from the early 70s. So these organisms are a little bit bigger, and we're really just starting to learn more about how they act in the ocean. And together, these two groups of nitrifiers are the dominant chemolithoautotrophs in the global ocean. As Rebecca mentioned in the introductory talk, they can make up 30 to 40% of cells in the deep ocean. So on the right-hand side are some of the data that prove this. This is a transect from my lab in the Pacific Ocean, crossing the equator, some data from Gerhard Herndl's lab in the Atlantic along the AMT. And we're showing here down to 1,000 meters, the abundance of these organisms as measured by the abundance of one of the key genes in their genomes that facilitate this ammonia oxidation reaction. So they can reach up to 10 to the 7th cells per liter of water. And it's interesting to see how their abundance varies through the mesopelagic ocean. And they're particularly abundant in areas of high export, which we can see around the equator in both the Pacific and the Atlantic. And so because of their abundance, I have long argued that if you really want to understand what's going on in the mesopelagic, you have to understand what's going on with nitrifiers. And I've been arguing this for so long that in fact, it was the topic of my OCB talk that I gave over 11 years ago, trying to think about how we could study nitrifiers in the deep ocean. And one of the last slides that I gave in that talk was a bit of a thought experiment. So again, this is from my talk in 2014, trying to reconcile for myself some measurements that folks were making using radiocarbon uptake of bicarbonate in the deep ocean. And these are very hard-won data. I don't want to downplay how hard it is to make these measurements. But estimates of carbon fixation in the deep ocean were coming up around 0.8 petagrams per year of carbon. And this seemed like a really high number to me. And I'll say at this time, when people talked about carbon fixation in the ocean, they were using that somewhat equivalently with chemoautotrophy, which probably shouldn't be the case now. But in any case, when I saw these numbers of carbon fixation at the time, being someone who studied nitrifiers, which should be the dominant chemoautotrophs in the ocean, that caused me to do sort of a back of the envelope calculation. So I took my favorite measurement of carbon export at the time, which had been published that year, took an estimate of the carbon to nitrogen ratio of that sinking organic matter to come up with a number for nitrogen export, and said, okay, if we had that much nitrogen entering the mesopelagic ocean, how much carbon fixation could we reasonably support with that much nitrogen? And to do that, you need a number of how much carbon ammonia oxidizers can fix for a given amount of nitrogen. And the only number available at that time was for an organism called Nitrosopomonas maritimus. And an estimate that I pulled from this paper was about 1 to 19. So ammonia oxidizing archaea needed to oxidize about 19 moles of ammonia in order to fix 1 mole of carbon. So with those 3 numbers, I did a back of the envelope calculation that you could only get about 0.05 petagrams of carbon fixed by AOA, ammonia oxidizing archaea, and you could only get about 0.01 petagrams of carbon fixed by nitrite oxidizing. And so that's only about 7% of the number proposed from these bottle experiments. And so looking at that, I thought, well, that might be a nice thing to spend the next 11 years on. And I thought, well, I can't really contribute a whole lot to improving our estimates of carbon export in the ocean. But one thing that I can probably contribute something to is the physiology of these organisms and whether or not this 1 to 19 number is actually right. So if we think about that problem in a prettier, more graphically oriented sense, and we think about ammonia oxidation, so these ammonia oxidizer, this is an ammonia oxidizing cell. As it's oxidizing ammonia to nitrite, how much bicarbonate can it take up for every mole of nitrogen it pushes through this system? And I also like to mention that these experiments aren't as straightforward as we might like them to be. This is a picture of exponential phase cultures of these nitrifiers. And if that looks a lot like water to you, it really does look a whole lot like water. We can't measure their growth using optical density, which is something you would use if you were working with an organism like E. coli. We can't centrifuge them very easily to get cell pellets. So they can be pretty difficult to work with. Not to mention that their growth rates are pretty slow, so every individual experiment takes a long time. So a real winning combination when you're trying to recruit people to your lab to work on these topics. But a fearless person whose data I'm really going to feature very heavily in this talk is Dr. Barbara Bayer, she was a postdoc in my group and now has her own lab at the University of Vienna. And she did a lot of really careful experiments, many of them during the pandemic, which was a really hard time to be in the lab, trying to get at this question of what is the carbon fixation efficiency of nitrifiers that we know live in the ocean. So here's an example of one of those experiments tracking an ammonia oxidizing archaea. We can track its growth either with nitrite, which is shown on this left vertical axis here, and the black dots, so an exponential increase in nitrite as they grow. And that occurs coincident with an exponential increase in cells. So as a just general overview of how she conducted these experiments, she added 14C labeled, radiocarbon labeled bicarbonate at the green arrow, and then collected the cells at each of the red arrows to get an estimate of how much carbon had been fixed for a given amount of nitrogen that had been oxidized at that time. And this is a summary graph of all those experiments. And so along the horizontal axis here are different strains of these nitrifying organisms. On the left in blue are the ammonia oxidizing archaea. Those are the dominant ammonia oxidizers in most of the ocean. She did a few experiments with ammonia oxidizing bacteria, which occur in sediments and are generally more common in terrestrial environments, and nitrite oxidizing bacteria shown in green. And over here on the left is the ratio of the amount of carbon fixed for nitrogen oxidized in mole per mole ratio. And so she found that this ratio for the ammonia oxidizing archaea we tested was about 0.1. So that's much, much higher than the ammonia oxidizing bacteria. And we think that that's just due to a function of their carbon fixation pathway. Ammonia oxidation yields a lot more free energy than nitrite oxidation. So the ammonia oxidizers are always going to fix more carbon for every mole of nitrogen that they oxidize than the nitrite oxidizing bacteria. So with Barbara's work, we could revisit that question that I posed earlier, look at this estimate of 0.8 petagrams a year and see how we could refine these numbers. So in total, Barbara found that the AOA are a bit more efficient at fixing carbon than reported just from these first reports of that single ammonia oxidizing strain. And the ratio, which is basically just the reciprocal of the number I showed in the last plot, is about 1 to 11. So for every 11 moles of ammonia that gets oxidized, 1 mole of carbon can get fixed. And because they're more efficient, we can revise those numbers a little bit higher that I showed the first time I showed this slide to about 0.1 petagrams a year of carbon fixed by the AOA and about 0.4 petagrams of carbon per year fixed by the nitrite oxidizing bacteria. But even with those upward revisions, we're still now only at about 16% of this estimate that we got from bottle experiments of DIC uptake in the deep ocean. So those are all lab experiments, and it's nice to have a way to kind of ground truth some of this in the field. And so Barbara came up with a very clever way to do that by probing some of the literature on terrestrial ammonia oxidation. And so what we wanted to do is find a way to specifically isolate the contribution of ammonia oxidizers when doing an experiment like what I'm showing here on the left. And so Barbara read about an inhibitor called phenylacetylene that had been used in terrestrial settings with soil microorganisms to inhibit nitrification. That's actually a really economically valuable thing to be able to do if you work in an agricultural setting to inhibit nitrification. And so the first thing we needed to do before we took this to sea was to show that you can, in fact, specifically inhibit ammonia oxidation by adding this inhibitor. So one of those experiments is shown on the left here, nitrite production in the control in the dark black line, and then different concentrations of phenylacetylene, abbreviated as PA. So you get a little bit of inhibition with 1 micromolar of phenylacetylene, and adding 5 micromolar totally knocks out the activity of ammonia oxidizing archaea. It also knocks out the activity of ammonia oxidizing bacteria. So these bar charts over on the right are showing carbon fixation in the absence, that's the minus PA, or in the presence plus PA of this inhibitor. So for both the ammonia oxidizers, the inhibitor totally knocks out their activity, and it doesn't do anything to the activity of the second half of nitrification, nitrite oxidation. So this is a nitrite oxidizing bacteria. Those two bars are the same. So we know that this inhibitor doesn't inhibit nitrite oxidizers. So to take this inhibition assay into the field, we took advantage of 2 cruises, one led by Anela Choy off the coast of California, shown in the green dots, and one in the eastern equatorial Pacific, led by myself and Max Saito. And this is just a general overview of what nitrification rates look like in the water column, if you're not familiar. In coastal areas, like up here, what we're showing is station 1, that's the upper left plot. In this graph, we get rates of nitrification around 60 nanomolar per day, and they're the highest just below the euphotic zone. So the gray dashed line on all these plots shows the depth of the euphotic zone. Further offshore, for example, like at station 7, in the equatorial South Pacific, we see rates more along the 20 nanomolar per day. So once we were at sea, Barbara also wanted to make sure that adding this inhibitor didn't impact other microbial processes. So we were measuring heterotrophic bacterial production as well, using leucine incorporation. And so that's shown on the vertical axis of these plots on the left, using the same color scheme I showed before, in the absence of the inhibitor with the inhibitor of nitrification. And so these bars are the same height. So that means that adding phenylacetylene doesn't impact heterotrophic production. We're also a little bit worried that the solvent that you have to dissolve this inhibitor in, DMSO, could inhibit bacterial production, and it doesn't appear to do that either. So that's great news. So over here on the right is what we actually want to use the inhibitor for and see its impact on inorganic carbon fixation. So here you can see, just as an example from 2 different stations, what happens, what a carbon fixation rate that you measure without the inhibitor and with the inhibitor. So that difference is what we can attribute specifically to ammonia oxidizers. So this is what that looks like in profile format. And in black is the total DIC fixation through the water column, and in blue is the DIC fixation less the contribution from ammonia oxidizers, so the rate that we measured in the presence of this inhibitor. And you can see that these profiles are lying pretty much right on top of each other. And only in some cases, right around the base of the euphotic zone, do we see a significant knockdown of the rate when we add this inhibitor. So what that says to us is that a whole lot of the DIC fixation that's measured when you add 14C bicarbonate is not really what I would consider true autotrophy. So if we look at that in a more budgeting sense, I know this is a pretty busy table. On the first row of this table is the total measured dark DIC fixation from adding 14C bicarbonate. And then the second row is what we calculated as the contribution from ammonia oxidation using our inhibitor method. We used a couple of other methods to estimate what we thought the contribution of nitrite and sulfur oxidation was to this DIC fixation. And the bottom line is we have to invoke a whole lot of heterotrophic inorganic carbon uptake to get anywhere close to what we see as the total DIC fixation. So again, back to our summary, when we go to the field and use inhibitors to measure the contribution of nitrifiers, we can get a little bit higher than what our geochemical mass balance budgets were giving us, but it's still in that same range of 11% up to, in some locations, 60% of measured mesopelagic carbon fixation could be attributed to nitrification. So the heterotrophs are contributing a lot to what we measure when we measure carbon fixation in this way. So the next thing that we wanted to ask is, we know that when photoautotrophic organisms take up inorganic carbon, that they release some of that as organic carbon, which is a really important carbon source for microbes in the surface ocean. We wanted to know if a similar thing was happening in the deep ocean with these autotrophic organisms. So at the same time that Barbara was measuring her carbon fixation efficiencies, she was also measuring how much of that carbon was being released as dissolved organic carbon, or DOC. So along the x-axis here, we have those same strains of nitrifying organisms that I showed you the carbon fixation efficiencies for. except for now on the vertical axis, I'm showing the percent of the fixed carbon that is then again released as dissolved organic carbon, in this case into the surrounding media, because these are laboratory experiments. So for the ammonia oxidizers, somewhere between 5 and 12% of the carbon that they fixed is re-released into the surrounding media. For nitrite oxidizers, it's a little bit higher across the board. About 12% of the carbon that they fixed is again released. Barbara confirmed that this is something that happens, that growing cells are doing. This isn't just something they do when they die. And another thing that I think is really interesting is that the amount of DOC released varies even in closely related species of organisms. So it really is something that we need to know on a species level. So what are these compounds? Barbara had a really great paper that came out earlier this year in the ISME journal about what some of these compounds are, with some great stories about how the compounds are shared. Just to give you a little taste of that, on the x-axis here are a variety of different vitamins, and everywhere you see a bar means that we detected that metabolite in the surrounding seawater and when these organisms were growing. And this huge peak that you can see is a compound called DMB, which is the lower ligand of cobalamin, which forms vitamin B12. So these organisms are releasing really high amounts of precursors for vitamin biosynthesis that could be used by other members of the microbial community, as well as a variety of other vitamins and amino acids. So a lot of the DOC that these organisms are releasing is not recalcitrant DOC, it's DOC that should be easily and readily available to other members of the microbial community. So we often think about this classic version of the microbial loop where photoautotrophic phytoplankton in the surface ocean that are using the sun's energy to fix carbon release dissolved organic carbon that fuels a heterotrophic microbial loop. And we think a similar thing is happening with chemoautotrophes in the mesopelagic ocean, that chemoautotrophes that fix DOC release dissolved organic carbon that can feed heterotrophic bacteria with very labile dissolved organic carbon compounds. But probably now we also need to add an arrow from CO2 directly to the heterotrophic bacteria because we know they're important for carbon dioxide fixation there. So just in summary of the story I told you today, about 20% of the depth integrated DIC fixation we could attribute to ammonia oxidation. And if we look at nitrification as a whole, including nitrite oxidation, the depth integrated contribution of those organisms was between 11 and 60%. And that about 12%, up to 12% of the fixed carbon from these chemoautotrophes can be re-released and as very labile compounds, including vitamins, vitamin precursors, and amino acids. So a huge thank you to the National Science Foundation for funding the oceanographic cruises that facilitated all of this work. And a big thank you to the Austrian National Science Foundation, the FWF, that supported Barbara's fellowship. The metabolic work, or the metabolome work, was done in collaboration with the Joint Genome Institute, the JGI, and the Moore Foundation, the Simons Foundation have been great supporters of the very slow and careful cultivation work that we do in our lab that is really can be hard to get supported in other ways. So a big thank you to them. And of course, a huge thank you to Barbara and all the great work that she did and all the great collaborators that helped contribute to this work. Thanks so much.
Thanks. That was great, Alyson. Thank you so much. We have a few minutes here for questions. If anyone has some, again, you can go ahead and raise your hand or just unmute and go ahead and ask or put in the chat.
Are you going to monitor the chat, Randie, or should I?
Yeah, I'll monitor it just because I think that's easier. One comment just from Phoebe. Great progress from your 2014 talk. It seems like the last 11 years are well spent. I agree. That was amazing.
Yeah, a little more gray hair than that talk. But...
Don't we all? Yeah. Let's see. There's one question from Alex in the chat. He said, great talk. Such elegant work. Have there been any measurements made of viral turnover of nitrifiers in any ecosystem, and any sense for how that release of DOC compares to what Barbara measured?
Yeah, that's a fun question. As you probably know, those are really challenging experiments to do, period, much less to do in the mesopelagic ocean. I'm aware of one paper that tried to estimate grazing rates on AOA, but I'm not sure of any estimates specifically of viral turnover. So I apologize to anyone on the call whose work I'm ignoring, but please chime in if you have measured archaeal viral turnover in the deep ocean. I would say that I guess any general estimate of microbial viral turnover in the deep ocean probably applies. We don't have any reason to believe that these organisms are particularly resistant to viruses. So for whatever that's worth.
Carly has her hand raised.
Related to mixotrophy. So I liked your little loop, but since we do know some of these are also mixotrophs, do you think some of could they be doing some of this to themselves or for themselves, or do you think there is some play of figuring out how much these are actually acting, of taking up the organic carbon themselves?
That's a great question. So I think that for the AOA, there actually is pretty little evidence that there's any organic carbon usage by the ammonia oxidizing archaea. No one that I know of really has looked at whether or not nitrite oxidizers, which are pretty well documented, there are a lot of mixotrophic strains in culture, whether or not they may also be doing that. We have a little data on one small carbon compound that we think could be involved in some nitrite oxidizer metabolism, but that would be a fun question to investigate with some of the tools that Anne uses in her lab, because I think you might need fish to look at that.
Yeah, go for it, Emily.
Okay, thanks for your talk, Alyson. I was wondering if, first, why do you think the autotrophs release all of these really nice compounds that they might also need? And also what, maybe if you've looked into what fraction of the heterotrophic need for those vitamins, maybe that is providing. I'm curious about that.
So great question. So to answer the first part of the question, why are they doing this? I guess I wouldn't necessarily presume it's something they're trying to do. We do think a lot of this release is a passive process and not necessarily something that they're actively pumping out, with a few exceptions. So in Barbara's paper, we did talk about some interesting stories where we also did a bunch of co-culture experiments where we grew these organisms in the presence of heterotrophic bacteria, and we saw an increase in the production of some metabolites in the presence of the heterotroph, and some of those were vitamins, which was pretty interesting to think about. So I think the jury's still out. It's a fun thing that we're actively working on to see whether we can grow some heterotrophs on basically the spent media from these organisms.
Let's see. There's another question in the chat from Bob Anderson. He said, do AOA have a competitive advantage because they fix carbon per mole of ammonia oxidized? And if so, why are there any AOB left in the ocean? I think I'm saying.
Yeah, that's a great question. Thanks for your question. There really aren't very many AOB in the ocean at all. There's some in sediments. That seems to be where they can have a little bit of a advantage, where there's higher fluxes of ammonia from organic matter breaking down. There also may be some ammonia oxidizing bacteria in the guts of marine organisms, but in terms of the pelagic water column, there basically are not any ammonia oxidizing bacteria. So you're right, they have a very clear competitive advantage.
Yeah, okay, great. I think Maria has her hand raised, if you don't mind unmuting.
Hi, Masha.
Hi, Al. So good to see you.
It's good to see you. Thanks for coming.
Amazing talk and amazing work, but I have a question about carbon rather than nitrogen. What energy do heterotrophs use for CO2 fixation? Is the energy derived from respiration? And if so, are they just spinning their wheels, or do you have any thoughts on this?
Yeah, the energy comes from organic carbon respiration in the heterotrophic CO2 uptake.
Do you have any thoughts why they fix CO2 then?
I mean, it's just bicarbonate. Part of those biochemical pathways involves the incorporation of bicarbonate.
So there is no advantage to that.
Right. So yeah, it's not necessarily like they're
All right.
Say this. Yeah, like the evolution, the biochemical pathway to like add a carboxylase like onto the end of your molecule, it incorporates a bicarbonate. So what I guess you could have asked a question, you know, was that originating from internally from their cell, from their own respiration, or is it something they're taking up externally? And it seems like from these rate measurements, it's something they're accessing externally.
Thank you.
Yeah.
Another question in the chat from Norm. Do AOA just use free dissolved ammonium ions, or do they use, say, ammonium residues on amino acids or peptides?
That's a really great question. So as far as we know, the enzyme, the ammonia monooxygenase enzyme acts on ammonia. So if they are accessing any other compound that contains an amine group, they need to cleave that off and generate ammonia. The transporter that they all have, most of them have ammonium transporters, so they transport the ion into their cells, but that the enzyme acts on ammonia. There's bits of evidence, including some work that I did that Dekas's lab led, showing that they can apparently use some nitrogen-containing organic matter compounds, the most prevalent of which is urea. So urease is really common in a lot of these organisms. I think about a third of the mesopelagic AOA can use urea. That number might even be higher from some recent work in Anne's group. So the enzyme itself is acting on ammonia. The degree to which they can use other compounds to get that ammonia from, I guess, is still a little bit of a question.
Great. Thanks, Allison. There's a couple more questions in the chat, including one from Sarah, our next speaker, but maybe you can answer them there while we switch over. That was really awesome. Thank you so much.
Thank you.
I also put a question in the chat. We can talk about that later as well. This is really fun. I'm gonna throw in some extra comments just based on the talk so far. I first want to thank everybody at OCB for holding all this meeting and doing the virtual switchover, and of course the dark carbon cycling group. And I'm going to put my acknowledgements up front. I'm going to share data from lots of different projects that have involved ship time and lots of lab work. And so I want to acknowledge all the folks that have helped assemble all of that. So today, I'm going to first so when they when they invited me for this session, I was really excited because I love the idea of going up the food web. Like this is the theme that Randie and Anne introduced today. And so my role is kind of thinking about carbon fixation in the dark, but also what's next and what happens next, which I think is one of the questions that was put in the chat earlier too. And I'm thinking primarily of the first consumers, those that might be involved in recycling that newly fixed carbon and are probably doing some other stuff as well, which I'll get to. And I'm going to talk about that. I definitely use this talk as a way to do a thought experiment and think about my work and how I can, what I want to do next, and maybe how I can get back into the lab and do some experiments to kind of extrapolate of what I've seen, specifically from some prior experiments I've run at deep sea hydrothermal vents. So I'll show the vent work as a little vignette to kind of introduce all of that. I, of course, started with a really overly arrowed and kind of food web depiction of kind of how I was to kind of draw one of these by myself, and I put as many arrows as possible in here. And of course, we're kind of thinking more about the dark carbon fixation. So I kind of removed this initial photoautotrophy method, but I'm still including phytoplankton because as has been stated, they're still contributing a lot to the sinking material and the sequestration of carbon, but now we have maybe a greater role that compared to the euphotic zone for things that are kind of responsible for that dark carbon fixation. And where I'm gonna take us today, or what I'm going to share today, is thinking about the heterotrophic protists in this. So thinking about the microbial eukaryotes as the maybe some of the first responders to that available organic carbon in a way. And I have a lot of questions. I have more questions, I think, that I ended up with in this talk. But I'm going to talk about when I think about these question marks here. What do I mean by that? The immediate one is phagotrophy, the role that microbes might play in heterotrophy and actually consuming prey. Okay, so we think of heterotrophy where a ciliate is filter feeding, or we actually do active hunting, finding a prey cell and actually consuming it, phagocytizing that prey, shown here with a burger, and then actually gaining energy. But I wanted to expand upon this with some of the data that I have today. And so the first one is parasitism, where a cell will link up with another. A lot of the microbial eukaryotes can be parasites of larger metazoan, other microbial eukaryotes or protists, and they'll typically attach, get inside the cell, and then parasitize that cell. And that's a big role, or I think it plays a big part in mortality that microbial eukaryotes can impart on the carbon cycle. Another one is symbiosis, where again, cells will form a linkage, and then maybe that relationship is going to be mutual, maybe it's commensal, maybe it's more complicated. I think parasitism could be considered kind of under the umbrella of symbiosis. And I also acknowledge that symbiosis may not be what one might think of as a conventional trophic mode, but it's also why I asked the question of Alyson earlier, because if these organisms are involved in symbiotic relationships, so there's more symbiosis, especially between bacteria and archaea and eukaryotes, maybe there's other ways that carbon is being packaged and processed and regenerated and delivered to the community. And so that's something to consider. And then I'm going to keep going here with my emoji-based definitions. Another one is saprotrophy, when we have these microbes that are really kind of dependent on decomposing dead, decaying material, right? We have a lot of sinking material, and there's micro eukaryotes that will take up that material to kind of consider a chemoheterotroph in a way. Similarly, we have an osmotrophic method of nutrient generation, and that's going to be where a cell is going to use osmosis to actually absorb those dissolved material. And then another one is mesocytosis. So I've kind of clearly gone over the deep end on thinking about all the different trophic modes of protists and what they might be doing in the dark. And mesocytosis is one that is more of like a cell gaining energy by prey cell perforation, or actually just taking a bite of another cell and actually like slurping up cytoplasm. So it's called like a cellular vampire in a way. So these are my depictions of that. Now, one of the reasons I made my overly complicated arrow food web before, instead of grabbing one and citing some of the really nicely generated ones from a paper, is that I wanted to try to add all of these components to my food web. And I really, at reiterating what people have said, and really just fueling what else is happening in the microbial loop here in the dark, contributing to sinking material, regenerating DOC, repackaging carbon. There's clearly these trophic modes. If we were able to get rates and things from rates and understand what's happening with each of these interactions and trophic modes that I've just described, we'd have a lot more understanding of that conversion between DOC and POC, how much labile carbon is being produced, what microbe interactions actually influence essentially the tastiness of that carbon. And I think that these are all kind of components that remain unconstrained. And it will ultimately influence the biological carbon pump. So that's kind of the introduction of why we should care about some of these maybe lesser known trophic modes in a way. And I think the way that I want to introduce this, or kind of get into this a little bit further, is looking at the hydrothermal vent environment. Okay, so this is a little bit of an exercise. This is all work done at deep sea hydrothermal vents, right? Where, as a reminder, this is a food web that's completely independent from sunlight, and it's primarily influenced or fueled by that chemosynthetic bacteria and archaea that are interacting with geothermally charged seawater that's subsurface seawater that's coming up very quickly. And so the reason I wanted to study this is that there was this beautiful gradient of what's happening right where that diffuse vent fluid is coming up, and you can access that diffuse vent fluid and think about what's happening in that chemosynthetically powered food web and then compare it right away to what's happening in the deep sea. Okay, so that's kind of where a lot of this project is, kind of the setting of a lot of these projects so far. And so my role here was to, and as a reference, I was targeting all low temperature diffuse vent fluid. So this is going to range, this is going to be less than 100 degrees Celsius. So we were looking at vents that kind of look like these pictures on the right. These are actually blue ciliate mats of the sessile ciliates, another trophic mode that I won't get into now, and not necessarily these black smokers, right, in terms of temperature. So as you might guess, my role was to really look at how the primary consumers, the microbial eukaryotes, what they're doing in this hydrothermal vent food web. There wasn't that much data when I first started some of these experiments on this, and so it was a interesting approach. I had these hypotheses based on using mining old metaomic data to see if there were signatures of protists even at the vent sites themselves. So I'm going to show some work from several cruises that took me to the Northeast Pacific, and then also down to the 2 vent fields within the Mid-Cayman Rise, Picard and Von Dam down here. And at each of these sites, like I mentioned, I kind of considered this gradient. So I took samples and did incubations, really trying to compare the local diffuse vent fluid to the plume where you still detected some hydrothermal vent fluid, and then the background where you were kind of isolated from the hydrothermal vent signal. And so I really want to juxtapose these different environments. So today I'm gonna do that again. I'm gonna make it slightly simpler and look at just the background and vent fluid and kind of do my DIY food web here. And I've tried to compile some prior data, and I'll talk about what I missed, what assumptions I've made, of course, as I go along. First, these light blue rectangles are my new dark carbon black box representation. Okay, we do have estimates of chemosynthetic primary production, and I know we've also have some estimates of the dark carbon fixation. Okay, but this is going to stay as a black box because it's showing that these still we need more estimates. We need to constrain this more. And that's kind of why we're all here today. But first, when I take these samples across this gradient, of course, I want to isolate and target the prokaryote population, the bacteria and archaea, the chemosynthesizers. And so we're doing cell counts, and I can convert this into a standing stock into micrograms of carbon per liter. So when we're comparing what's happening in the background compared to the vent fluid, we're going to see that the stuff from the diffuse vent fluid is going to be much more elevated in the background. One thing that I know that I've missed is, and that was mentioned by one of Anne's students, I believe, was talking about the carbon conversion rates for cells. I'm using one version of a carbon conversion rate, and I've played around with them. And I think that that's something that we should play around with and consider when we look at these different environment types. The next, going up to my DIY food web, this first one is going to be grazing rates. And this is where I was running my incubation. This was kind of my primary goal, to quantify this exchange of carbon. And to do this, I ran a series of fluorescently labeled bacteria experiments where the cell takes up the fluorescently labeled bacteria that you provide it. Okay, so that's kind of what you can see here, with the cell actually consuming some of that prey that basically fluorescently labeled bait tray that you've provided. And that gives us these grazing rates. And so I have these for Gorder Ridge and then the 2 sites in Mid-Cayman Rise area where we have the cells consumed per mil per hour, okay? And across 3 different vent fields, we consistently see that the grazing rate is higher at the vent site. This makes sense. There's a chemosynthetic ray buffet at the vent site. There's going to be more biological diversity kind of localized to the vent. We already know this. And so this makes sense that we're finding this. And I'm going to just chime in with one tiny comment. I did run some of the Picard and Von Dam experiments under pressure. It's really hard to run these. It's really hard to run any experiment under pressure and retain it. But I have some that were run under pressure from Picard and Von Dam showing that you do get higher rates, but they're just not enough to statistically kind of make any large generalizations about it. But it's just really hard to do things at that kind of pressure. But I'm actually now taking those grazing rates from the cells consumed per mil per hour. I'm using the same carbon conversion factor that I used when I estimated the prokaryote biomass, and now looking at the amount of carbon that might be taken up by that Protists community. Okay, and so we see something like a pretty decent amount might be taken up from the Protists in the vents, and then less so in the outside, but outside of the vent site. But again, this is just kind of one value, and I'm averaging across lots of experiments here. And then so to continue my food web here, I also, during the grazing experiments themselves, you end up getting an idea of how many eukaryotes are in your system. And I was able to get the eukaryote cells per mil. The overall abundance of these cells is lower than what we see with the bacteria archaea. But these are larger cells, okay? And so when you use, and actually when I visualize them and count them, I actually measure around the cell in the microscope. I calculate a biovolume, and then I use a carbon conversion estimate that assumes a mixed microbial eukaryote community, but excludes diatoms to get this carbon conversion value. So again, using field and field standard carbon conversion value, but again, using one for my whole system here. And the amount of carbon that this pool represents is something that I think is fascinating, and one thing that I want to continue thinking about, and because it kind of has implications for the rest of the food web, because this is a pretty decent amount of carbon when you think about how much the next trophic level might be looking for in these environments. So that's kind of my initial DIY food web. Things that I'm missing. Some of the big take-home messages first. All right, if you modify and start messing with and changing the parameters on the initial chemoautotrophy and some of this, then you're going to see other things propagate through the food web. And so that's kind of, again, one thing that I'm hoping to learn more from this group, and maybe think about how we can constrain each of these values a lot better. I mentioned one thing that's missing the mark is definitely the range of carbon per cell that we're estimating. I think that there's a big range here. And the other thing that I've found is that when I did the biovolume calculations for the eukaryotes, is that they were overall smaller outside of the vent compared to inside of the vent. And so we know that we have more cells within the vent site, but they're also larger. And so the eukaryote cells themselves are larger than bacteria and archaea, but the relative range of the species, size range of those species of eukaryotes is also going to be different depending on the location. And again, the reason I'm comparing this vent site to the background deep seawater is that it's a way to kind of show how, if we know that the deep sea is also pretty patchy, and there's going to be this mosaic of things that have a hot spot like the vent site compared to outside. We've seen things from the halocline from Orca Basin that was shown earlier, and I've seen other grazing rates show similar increases compared to the background deep sea water across redox clines. And that's something else that we need to take into account when we're starting to get larger, we're trying to model, I guess, or get a bigger depiction of what's going on in the dark ocean. Okay, and another thing that I keep thinking about is this assimilation efficiency. Okay, like we know that these organisms might be taking up this material or interacting with this material, but how much of that, like, where does that actually go inside the cell? So I'll get to some of those things in a second. But kind of continuing on with my vent example, the other thing that I didn't really address here is I just showed you all my emoji defined other trophic modes that could be happening in the dark. And I'm really only showing grazing here, the heterotrophic modes of nutrition. And so paired with all these samples, we have also done 18S to kind of get an idea of who's present in these environments. And these tiles represent what's happening in the background, plume in the vent. And each tile, the square tile and the individual colors inside are kind of a different version of a relative abundance bar plot for metabarcoding data. And the colors are represented by the different Protist taxonomic groups. And so I'm kind of showing from left to right, background to vent, and then from 4 different sites here. And really the main take home is that we see a shift. So we have a more diverse vent specific signature within these sites compared to the background deep sea. But more related to this talk and kind of where I'm heading next is when we start opening up and thinking about the species that are represented in all these taxa, there's a lot of different trophic modes that are established, okay? And so we've been doing like deep dives to try to understand what of these organisms are capable of a lot of these different trophic modes. So we've established a lot of that heterotrophy or the phagotrophic modes of nutrition. We've quantified that. We're looking for genes that are specific to parasitism, might show some kind of membrane transport with or sharing of pathways with symbiosis and osmotrophy, mysocytosis, and even saprotrophy. And so really, I think that this is kind of what's next right now. So in addition to this metabarcoding data, we're now processing a lot of the metatranscriptome data from this and trying to understand how we can maybe separate out these trophic roles from what we see in the community composition or from the whole community. This is one example of it. What I've done here is take our metatranscriptome data. Okay, so a little bit of background on that is that it's messenger RNA selected, poly-A tail selected, and then we're using a euchromatic to process it, and we've annotated it with PFAM, Go, and KEGG databases. And I'm just clustering at a really high low taxonomic resolution. So have ciliates all included here. And we're seeing some trends. This is only vent data across different vent fields. Okay, so one thing that's interesting is that there's some grouping by taxa, but the vent sites themselves aren't really showing a key pattern here. One of the other things that I'm not showing is that when we do include cell counts, temperature, pH, and other parameters that we collect with the geochemical parameters we collect with our vent fluid, we do see that a lot of that can explain up to 80% of the variants that we see in our transcript signatures. And so we're digging into each of these trophic modes to kind of understand if any of them might be related to what's happening in the environment or any trends that might appear there. And then, so this is one approach. And one of the things I did next is also one of the things that's kind of paired with this is that each of these trophic modes, I'm trying to look at the carbon processing pathways that we know exist, and like maybe with the KEGG pathways and the KEGG orthology, and pull out things that might be associated with like the different phagotrophic modes of nutrition. I mentioned transporters with certain symbiotic relationships, and there's a lot of membrane interactions with respect to endocytosis and breaking up of membranes, or a parasite having to protect its own membrane to enter a host, and pulling out individual genes that might be associated with those, and trying to see if those are shared between things that I'm hypothesizing to be parasitic or have symbiotic relationships with other organisms. Up and coming is definitely going to, this is an example, next step, where I've taken more of an unsupervised clustering approach to look at the transcript profiles across lots of different protist taxa here. And we're seeing that there's some patterns that might start appearing. I think that this is one of the first steps where we're going to start reevaluating how we can try to predict the taxonomic groups, predict what trophic mode they're a part of, and see if we can use unsupervised machine learning approaches to start placing them on that spectrum of phototrophy to heterotrophy, or other trophic modes of if they are showing some signs of saprotrophic modes of nutrition or something else. And so this is one way we're trying to experiment with that work. And let's see, I know I'm kind of running out of time, but one other thing I want to point out is that when I have these individual clusters that form from the transcripts I've kind of put into this, I see a lot of taxa that have representation across the different clusters, and some that are kind of isolated to one. And as we go through this data, we're finding that some of these that are isolated to one or two are also isolated spatially throughout the vent site. And so we're thinking that there's this balance between the vent environment and also specialist strategies within some of these key taxonomic groups. And one hypothesis is that this is related to a lot of the Protists species that we think might be endemic to these vent sites. So that's another route that we're going in the future. So I will show this one more time, and I'm just kind of thinking about when I look at this overly pointy food web here, it's just important to consider all of the different relationships that the primary consumers might be having with the primary producers in this environment. There's predator-prey relationships that will also impact the community composition of some of the bacteria and archaea in the water column. And a lot of these processes kind of are I think of as primarily loss processes. They're contributing different compositions of labile carbon to the DOM pool, and they're contributing differently to how sinking material is being aggregated. Okay, I will go ahead and stop there because I believe I have run out of time. But I think that just one parting thought really quickly is that I think that this does matter, thinking about the dark carbon fixation, getting those quantified and constrained better, and then pairing that quantitative work with our qualitative work will help us kind of get to how we can pull out these individual trophic modes that I really am excited to pull out next. So I will end there and hopefully have some time for questions. Thank you.
Thanks so much, Sarah. Yeah, we have time for one question as we're getting ready to switch over to our lightning talks, and then there'll also be more time later in our kind of discussion period. But we have time for a question if anyone has one. Go ahead and put it in the chat or raise your hand. Feel free to unmute, or we're going to move into our lightning talk. Sarah, I really appreciate you giving us that perspective on this kind of consumption of the organic carbon that we're thinking about being fixed, that Alyson and Anne's group set us up for. That was really interesting. And I think we'll have good, fruitful discussions later as we start talking about the implications for carbon budgets. That was a great setup. If our two lightning talk speakers, Nicola and Hope, could go ahead and turn their videos on. Yeah, thanks, May. That'll get us ready for our 5-minute lightning talks.
Hi, everyone.
Great.
I'm Nicola Paul, and I'm currently a PhD student working with Alyson Santoro at UC Santa Barbara. And today I'll bring you through some of our work investigating microbial dynamics on sinking particles. So while there are several processes that contribute to flux attenuation, today we're just going to focus on the microbial contribution, and that's been shown to range from 7 to almost 30%. But that value depends on the region and the primary factors setting remineralization rates. So temperature, oxygen, or community composition, but the extent of their impact is largely unknown. And so links between microbial community structure and particle degradation have been observed both in the lab and in the environment. So for example, in the top figure here on the right, shifts in diversity were related to shifts in particle metabolism. And then in the bottom figure there, dominant genera on particles showed different metabolic capabilities on particles. However, being able to directly connect these shifts to in situ rate measurements proves to be pretty difficult. And so to try to get at this connection, we deployed sediment traps on surface tethered drifting arrays across contrasting ocean basins. And we co-deployed preservative containing pits and live respire traps, shown on the right side there again, which are these dual particle collector and incubator systems that allow us to measure particle-associated microbial respiration in situ. Additionally, we took DNA samples from both those poison traps and the respire traps for 16S amplicon sequencing. And we also measured bacterial production, cell counts, and DOC change, but I won't include those results today for the sake of time. From our deployments, we measured a wide range of POC fluxes, where our low flux regimes were measured in the Eastern tropical North and South Pacific. That might be a little bit cut off there. But then our high flux regimes were measured in the North Atlantic during the end of a diatom bloom, and then off the coast of California in the Santa Barbara Basin. Our carbon-specific respiration rates also varied pretty widely across regions, and then vertical trends even varied among deployments within the same region. So we want to think about how shifts in the community composition might relate to these measurements and contribute to that variability we're seeing. So we can start by looking at the overall community composition using this NMDS ordination plot, where each point represents the community on a particle sample. And we see these communities differ significantly by location. So then we can take it a step further and look at who's making up these communities and potentially contributing to our observed differences. And so looking at the level of amplicon sequence variants, or ASVs, which are unique sequences of DNA in the community, we have the ASV name on the y-axis. That might be a little cut off there, but each individual row is an ASV. And then on the x-axis, looking at relative abundances. And despite differences in the community we saw on the previous slide, chemoautotrophy consistently dominated on sinking particles across ocean basins. And we even saw some of the same ASVs present on particles across these contrasting regions. And those are the examples I've highlighted there. And this includes groups like Coelia, Pseudoalteromonas, and Olivibrio, which were present at all of our sites except the Santa Barbara Basin. So for these ASVs, we can also look at the change in relative abundance over the course of our incubation. And for several of the ones I highlighted on the previous slide, their relative abundances increased after incubation, indicating these taxa are actively growing on sinking particles. So then if we have some of the same ASVs growing on particles throughout the ocean, they could be good candidates to compare to our rate measurements and maybe some sort of indicator for particle remineralization. And so when we take two of these ASVs, these are both pseudoalteromonas that were found at almost all of our sites, we observed a positive correlation with our carbon-specific respiration rates from the respire traps. And these relationships only strengthened with the exclusion of a couple outliers. And so this demonstrates that changes in the community down to the ASV level could potentially serve as indicators for changes in remineralization across ocean basins. And so I'll just end on this summary slide with a few takeaway points. and also want to acknowledge the people and funding that made this possible. So thank you for listening, and feel free to email me with any questions.
Great. Thank you so much. We're going to have a couple minutes at the end, I think, so there should be time then for questions for both of our speakers. So I'm going to go ahead and move now to Dr. Hope Irene's talk, and then Nicola, we can at the end, if we have time for questions, folks can ask you some.
All right, well, thank you. As Randie mentioned, my name is Hope Irene's. I'm a postdoc with the U.S. Geological Survey at the Pacific Coastal Marine Science Center in Santa Cruz, California. And broadly, my research investigates organic carbon and storage and cycling in sediments that also host marine minerals. And today I'll be telling you about some of my work within hydrothermal systems that I've been working on with my supervisor, Nancy Prouty, as well as Amy Gertman and Pam Zorensky, all also here at USGS. And I'll note my work is just one piece of a much bigger puzzle of interdisciplinary and interagency work to try to understand these systems. So as I think many of us are aware, and we just chatted a little bit about, hydrothermal events are really unique environments with very specialized carbon cycle processes that could impact the global carbon cycle, including inorganic carbon fixation, primarily via chemosynthesis, as well as thermal alteration of DOC and sedimentary organic carbon, petroleum formation, and they may also host critical minerals within seafloor massive sulfides that could be of economic interest for potential mining. But baseline carbon cycling and storage at these systems is still relatively poorly understood, particularly as vents transition from active, such as those seen here in these pictures on the left, towards inactive, meaning we're no longer observing hydrothermal fluid flow. But inactive vents are also those most likely to be impacted by any potential mining disturbance and are relatively understudied compared to their active counterparts. So a goal of my work is to characterize baseline sediment organic carbon storage and cycling throughout diverse hydrothermal mineral systems that span this range of activity and temperature gradients. To do this, I've gone out with the USGS Global Seabed Minerals Resources Group led by Dr. Amy Gertman here at USGS in Santa Cruz. This map shows in yellow stars the two sites I'll be talking a bit about today. The Escanaba Trough is off the coast of Northern California. It's a sedimented, slow-spreading hydrothermal system. And the Galapagos Spreading Center has much more limited sediment coverage with an intermediate spreading rate. At both sites, we collected sediments via push coring, which you can see in these photos here at the base of active and inactive vents, as well as distal or background sediments. These push cores are about 20 centimeters, so we're really talking about the surface sediments here. And I first want to note that these two sites were very different in terms of total carbon storage, organic carbon source, the extent and evidence for thermal alteration. But for this brief talk today, I'll just highlight what we saw in common. So on the left here, I have a PCA of some of the data I measured, including carbon and nitrogen stable isotopes and various lipid biomarker indices. And in the top panel, you can see active sediments in purple. The inactive samples are in yellow, and the distal or background samples are in that blue color, with the variables used on the bottom panel. And the active sediments were really primarily distinguished by biological controls. So they had low carbon isotope values indicative of chemosynthesis, low nitrogen isotope values that corresponded to high pore water ammonium, and we attributed this to incomplete nitrogen utilization. And the only thermal alteration indicator that was consistent between the two sites were low pristane and phytame ratios. In contrast, the organic carbon in the inactive sediments very strongly resemble distal sediments, despite clear differences in the mineralogy and total carbon or carbonate, suggesting that there is hydrothermal material in those sediments. So I think this just really highlights the temporal nature of the organic carbon in the sediments that persists a little bit longer term after activity ceases. And then just one last brief plot here. This is specific to the Escanaba Trough, showing data using a tool called compound-specific isotope analysis of amino acids. So this is data based on the nitrogen isotope values of amino acids. And along the x-axis, we have just bulk nitrogen values. We found that those low nitrogen values in the active samples corresponded with high indicators of microbially reworked or resynthesized organic carbon, as well as high trophic positions. So kind of aligns well with some of what Sarah was showing us a little bit earlier, but suggests that we're seeing chemosynthetic organic carbon supporting more complex food webs, and specifically the organic carbon that's stored in the sediments. So if you're interested in learning more about this work, I'd encourage you to please check out our paper published this year in Chemical Geology. We also have a plain language video and summary on the USGS website that you can access from this QR code, and I'd be happy to chat more about it at a later date. Thank you.
Great, thank you so much. We have a couple minutes here before we have a scheduled break. So if anyone does have any questions for either of our lightning talk speakers, please feel free to put in the chat or raise your hand, etc. Okay, there's one in the chat from Clarissa, said great talk. They have a question. Do you think that the different communities cause the difference in respiration, or are they and the differences in rates the result of different particle compositions, like for example C to N or labile versus refractory, or conditions like temperature? So did you test for those factors?
Yeah, that's a great question, and we did look at both of those. Taken all of our sample regions together, there was no clear relationship with temperature. Breaking it down by location, for the most part, our mid to high latitude sites did not show a relationship with temperature. Maybe a slight positive relationship in our Eastern tropical Pacific sites, but we also only have a couple data points per deployment for those regions. Yeah, thinking about labile versus refractory is also a good question. Some of the data I showed from the North Atlantic and North Pacific were part of the NASA exports campaign, and so that targeted the North Pacific as a lower flux, more reworked particle environment versus diatom bloom in the North Atlantic, more labile material, and our carbon specific rates were much higher in the North Pacific, where the material was more reworked versus the North Atlantic. So definitely some contrasting thoughts there on what could be causing that. But yeah, we've been looking at some of these other components too.
That's great. Thank you so much. So we're gonna now continue to move up the food web, and we're gonna hear now from Stephen Haddock and Mbari, and he's gonna kick us off for the afternoon.
Thanks. So yeah, I would like to thank the organizers for bringing me into this meeting. I know a lot of the people in the attendees from way back when I was at UC Santa Barbara, but I haven't really interacted with them much. And after seeing the first talks, I'm having some crippling imposter syndrome. But I was thinking, if you can become the head of the Federal Emergency Management Agency without realizing that there's a hurricane season, then I might be okay for giving this talk. So I've been tasked with talking about the higher ups in the food chain, and I'm really gonna not distinguish between inorganic and organic carbon, because I think once you get to this point, particles and other marine snow would be some combination of those two sources. And plus, I think we really don't know that much about distinct specialized pathways for consuming them. So apologizing up front, I'm going to really just talk about carbon flux in general. And one of the things that we face when we're talking about this is, these schematic diagrams like this that are really useful for framing it. But if you look at this, the top 100 meters is drawn in this diagram like this. And then the next 3,700 meters are drawn in this little tiny section here. So there is a lot less focus, I think, or less knowledge about what happens in the deeper waters of the ocean than what happens up in the sunlit waters. And Anela and I wrote this review paper, and we were trying to think of different ways to kind of flip people's perception of the ocean a little bit. So this is a bathymetry map with the mid-Atlantic ridge here. And instead of draining all the water out of the ocean and showing the seafloor, what we did was we took the water and we raised it up above the surface of the earth to an equivalent height of what its depth was. And this gives you a different perspective on, things like the Marianas Trench. Instead of being a dark scour, is actually like a thin, sharp ridge, and the Hawaiian Islands become scour. So this just gives you a sense of how massive that volume of deep ocean is. And if you think about it numerically, you often hear about how the ocean is 2/3 of the Earth, but that is 2/3 of the area of the Earth. And since the average depth is nearly 4,000 meters, if you do an equal volume projection, you see that most of the biovolume on the Earth is ocean. And out of that biovolume, most of it is below 200 meters. So most of it is the deep sea. So we have this very, very large part of the biosphere, part of the planet, that is the deep ocean. And it's just a challenging place to study and to figure out what the processes are. But just by its sheer scale, it has a lot of importance in these dynamics of the things that we're interested in. So typical ways to study that are with nets. These are two different kinds of nets. This is actually the mock nest system, where you can do 10 samples at once. It's one of the most commonly used oceanographic tools for studying life in the deep. This is a different Tucker trawl it's got a single insulated cod end at the end, and this very long taper. And there's all kinds of nets in between, but these are some of the major tools that people use. And when you use those tools, you of course find what people think of as living in the ocean or in the deep ocean, which is a lot of crustaceans. And these are certainly important, and they're consumed by a lot of organisms, but they're not really the whole perspective. And so we use some other techniques to look at other parts of the ocean community. For surface waters, we can use scuba diving and collect things by hand into jars. And then for deeper waters, we can use remotely operated vehicles or human-occupied vehicles to collect. And when we use these other techniques, we see a different perspective on the ecosystem because we're able to sample the more fragile gelatinous components. And in particular for my talk today, there's a few groups of interest, which are these salps, polychaetes, the mollusks, and the what I call radiolarians, but they're basically these rhizarians, cercozoans. And so those are some major players in the food chain. They're not clearly not related to each other at all, but they've converged on this more transparent gelatinous morphology because they're living in this fully three-dimensional open ocean ecosystem. So in order to quantify the different perspectives that you get from applying these different methods, with Eric Tucson and I, a while ago, we got funding from the Census of Marine Life, and actually the Office of Naval Research donated some ship time. And we had 2 ships out at the same time in Monterey Bay. And we did some trawls with mock nests. We did trawls with the Tucker trawl, and then we did ROV video transects at the same time. And with the trawl samples, we took them on board. We sorted them out to morphospecies. We weighed them, we counted them. And we also took water samples at the same time so that we could get the microbial abundance. And we were able to compare then the results from those nets. So here you look at the mock nests. This light orange piece of the pie is the crustaceans. A darker orange piece of the pie is the shrimps. And you can see that the mock nest is mostly crustaceans. And then this blue wedge is the more gelatinous organisms. And the green ones are kind of the more hardy gelatinous things like heatignass, arrowworms, and salps. So with that Tucker trawl, we see that instead of being 2/3 of the pie, the crustacean part of the abundance was less than 1/4. And then there were much more of the gelatinous organisms in there. And of course, with the ROV, we have a lot of avoidance by crustaceans, and we see the more conspicuous jellies more easily. So you can get a completely inverted picture of what is living in the water column depending on which method you use. With these first two samples, we were able to quantify them, so we could get wet weight and then dry weight and then eventually carbon from all those bulk samples. And I'll just show you the carbon part of the equation. But again, with the Mach Ness sample from 750 meters, most of the carbon that we got was from those crustaceans and a very small fraction from the jellies. With the Tucker trawl, the crustaceans were maybe a third, and the combined crustaceans and fish were a little over half. And then the jellies started to make a more appreciable contribution to our biomass estimates. These are not on the same scale, though. So if you scale them to the amount of total biomass, that was just percentage composition. We retained less biomass in the Mach Ness samples because we were missing the jelly fraction, and there was probably some avoidance in the fish fraction, as well as damage. So your estimate of the amount of carbon per volume from the nets is very different. And I think an important implication of this is that when we compare how much biomass is in the bacteria with the Mach Ness samples, so these are the same amount of bacteria, but just projected into that same per volume estimate. It's 55%, and this actually matches some of the numbers that you see in the literature of like how much of the biomass in the water is microbial or bacterial versus from animals and eukaryotes. And just with these two nets, neither, which is perfect, but you get a very different view of the community depending on the amount of biomass that you retrieve from a net. So I think this is an important caveat to a lot of the back of the envelope calculations that we made about how much carbon is in the ocean and how much carbon is in the deep sea, is that it really, your answer that you get is going to strongly depend on the methods that you're using to quantify that. So these organisms that we're looking at, they're not just living in isolation, they're eating each other. And again, with Anela, we did a study where if you just use gut contents or net caught specimens, you can get an idea of the food web with fish eating shrimp and kind of this portion of the food web. But with the ROV video, we saw much more numerous and diverse interactions between things. So these are coded by the tendrils are the color of the predator. So in this case, there was a jellyfish that ate 22 different kinds of prey. It's a jellyfish that preys largely on other jellies, but also will eat things like crustaceans, which were pretty obviously going to be the prey for a lot of our different organisms that we were seeing. One thing that's not in this, because this was based on video observations where we actually saw organism A eating organism B. One thing that's not in here is really the marine snow part of the equation. There is a little detritus circle here, but we couldn't really quantify that. But there are a bunch of different groups that I'll show in a little bit more detail that consume marine snow. And I think that's really a vector for getting very small sources of carbon into the much larger part of the food chain. It doesn't have to go through 3 or 4 intermediate steps to get from bacteria to a fish, for example, or to some zooplankton. And each of these tendrils is actually kind of an interesting story in itself. So this is one example where we had a comb jelly. We found this comb jelly that's still an undescribed species, but it specializes on eating this certain polychaete worm. And we've seen it actually basically catch them in situ. And you can see by the yellow gut of this polychaete, this is a detritivore, so it creates a mucus, a big mucus web and pulls in marine snow. And so all this brown is basically marine snow. Henk-Jan Hoving and others have suggested actually that even the vampire squid eats marine snow. My squid friends don't totally believe that. I mean, they believe it could be a part of the diet, but not that it's entirely subsists on consuming marine snow. So I would say that jury's a little bit out on that, but it certainly can catch particles and ingest them. So even something like a vampire squid. Some of the major players also are mollusks and pteropods. So these will again have a mucus filter that they use. Some of them are predatory, but these the shelled ones are often mucus feeders on detritus. And these also are of interest because they have inorganic carbon in the form of their calcium carbonate shells, which they, when they're swimming, so this is a heteropod, actually not a pteropod, but you can see that little conical shell that it's carrying around on the back of it. And this was a transect in the Gulf of California, Mexico. There were just tons of these shelled pteropods in the water column, and even in the oxygen minimum zone, where they would presumably be challenged by dissolution of their shells potentially. But they can be really abundant and important, both as herbivores and also as prey for other organisms. Another species that's pretty prominent in the marine snow consumption game are salps and endoleolids. And so these guys continuously pump water through, and they can get to be, if you say, oh, there's one salp in our sample, well, that one salp could have been 40 feet long or, 10 meters long and consisting of many, many of these zooids, all of which are continuously pumping water through and filtering out very, very small material, phytoplankton, and even marine snow. And then another one that has been studied somewhat lately by Bruce Robeson and historically by Debbie Steinberg and Per Flood and Russ Hopcroft are the larvaceans or appendicularians. This is the largest ones that we know of. The house can be, I don't know what size it is, the size of a large grapefruit or something. They have two filters that they make, and they pump water with their tail through, and you can see all this yellowish color again is material that they've accumulated and filtered from the water. And you can even see in the mouth the marine snow and particles that they are sucking in. And if you look under a microscope, this is some of Per Flood's results, but the mucus filter is really incredible. It looks like a piece of gauze or something. It's not just a random polysaccharide matrix. It's actually very uniformly laid out and can filter, probably, if not individual bacteria, certainly bacteria that are associated with some of these snow particles. And then one of my favorite groups are these things that are known by many names. So these are phaeodarians. They're cercozoans that we heard about in Sarah's talk. Historically, they were called radiolarians, but they're actually a distinct lineage from these other kinds of shallow water radiolarians. And so the shallow water ones got the name, and these guys have, now we call them rhizarians and cercozoans. And on Manu's talk a couple days ago, he showed another drawing from Haeckel where he was saying, we don't even know what their orientation is. How should we be presenting these? Because we don't know their orientation in the water column. And actually, for some of these now, we do know their orientation, which is they're oriented in all ways because they occur usually in these colonies of 8 attached to spheres or in an assemblage of 8. And this is about a centimeter and 1.2 centimeters across. So even though it's a protist or a microbe, it still can be pretty large, and they're very conspicuous in the water column. And the reason that they have that configuration is because they're optimized to gather marine snow. So they're slightly more buoyant. They're slightly more buoyant than the snow, so the snow sinks down past them. And all this whitish material is marine snow that's been accumulated by their mucus and also by the hooks on these long spines that they have. So if you look at one of those capsules that's isolated, this whole thing that's called the phaeodium is marine snow that it has ingested. And if you look at the autofluorescence of those, you can see this is not stained, but you can just see the diversity. There's some chlorophyll containing Protists in there probably, but also, there's all kinds of potential microbes and things that have been ingested by these radiolarians as part of their lifestyle. They also serve as a substrate. So this is a copepod that's Onsia. I think it's still called Onsia. It might be Pseudolabuckia, but these are copepods that don't swim freely in the water column. They crawl around on marine snow, but they also crawl around on radiolarians, and we've even found their eggs, that they've laid their eggs on the radiolarians. So again, as I mentioned, so these are the phaidarians, and these are the other things that are called radiolarians, not at all closely related to each other in these results. And then there's also forams, which are another similar. It's kind of like these are amoeba with a silica shell. These are forams are amoeba with a calcareous shell. And so we do see a pretty high abundance of these other large protists in the water column that may be more predatory than detritivores. So if you think that's just a sampling of many of the organisms that are found through the water column that eat marine snow. This is again from this review that Anela and I wrote, looking at the distribution of these different taxonomic groups from the surface to 4,000 meters based on the ROV video. So again, this is the macroscopic things large enough to see with a high def camera. But if you look at the composition of any one of these groups is going to change somewhat with depth. Like down here, the polychaetes are not the same species as they are in the middle. But if you look out of those, the ones that eat primarily marine snow or detritus, it's a pretty large proportion of the organisms down in the midwater that are going to be short-circuiting these trophic links by feeding directly upon marine snow. And so I think they have a high effect both on breaking up particles and putting them back into the water, or putting them into fecal pellets in some cases, and kind of shunting that carbon around in different ways. Those are the ones that we kind of know somewhat about. There are some other more speculative things that I think would be fun to investigate. So of course there's the oxygen minimum zone, which characterizes a lot of our coastal waters, and have this kind of fantasy project with Pete Gerges where we're trying to look at if there's any kind of chemosynthetic activity that happens in the oxygen minimum zone as a place where you get some potentially different chemical processes going on, probably through symbioses. But I don't know if anybody wants to like speculate about that with me later. I think that would be a fun thing to look at. So we're trying to take these from being just like vertically integrated measurements to actually being able to develop time series of them. And I had to show this result from Alex Jaffe when he did an internship in our lab before he's gone on to greater things. But we were looking at, more or less long time series, 20-year or so time series of different organisms in our data. And you can see that they sometimes are out of phase. So this siphonophore, these guys could both potentially be eating krill, but in this time period, the ctenophore was highly abundant and the siphonophore was less abundant than average. So one of the goals for looking at these kinds of data is to try to get longer term time series beyond just a simple snapshot. Another goal is to get to those smaller things. As I mentioned, the ROV video that we use a lot of this for is only good for things that you can see, maybe the size of a grape or larger. And this is a plankton microscope that was inspired by some of the Scripps imaging things, but it was built by Paul Roberts and Tom Affin. And we put it on these autonomous vehicles so that those can fly around. There's actually a couple out in the bay right now. You can send it on a mission and send it out for a week, and it will it will send you back a couple million images of plankton that it has captured. So we're trying to zoom into that smaller size fraction to really be able to get a better picture of what happens. And just as an example, this is a image from that microscope. So this is the medusa of the Velella that you see like washing up on shore. Those little rafts are actually the polyp phase, and this is the sexually mature like adult phase, but we can see those medusa with that little microscope. So thinking about carbon flux, I think it's also important in some other contexts, and maybe even more so recently, in the context of deep sea mining. And because deep sea mining, the process starts on the seafloor, and there's a lot of focus on the effects on the seafloor, but that's just the beginning, because once the particles are processed on the ship, they re-release the silt and the materials back into the water column, and it doesn't go all the way down. So you're creating this plume of somewhat sinking particles, but from what I understand now, it's all been so blenderized that it really creates this plume that doesn't sink out of the water column. And the effects on the organisms that I've been talking about are pretty pronounced. So things that eat marine snow, like those pteropods, they're going to be eating a large proportion of completely inorganic, like non-nutritive clay and sediment instead of what they're supposed to be eating. So I think as we think about these flex questions, it's good to keep an eye on developments in things like deep sea mining. And again, working with Anela, one of my favorite collaborators, we wrote an op-ed about this to try to draw attention. Don't ignore the water column when you're thinking about the effects of deep sea mining. And one last point is that I think it's important to create these carbon budgets, but for me, the animals that we work on, it's hard to quantify their value in petagrams of carbon, because they serve so many other roles in the ecosystem. And I think for people to care about funding research in these environments and for people to care about preserving them, we probably should have some other metric like emojis or something, to quantify the value of the animals that live there. That's it.
Thank you so much, Stephen. That was great. We have a couple minutes for some questions. If anyone has them, feel free to raise your hand or put in the chat. Those are beautiful images. Let's see. We have a question in the chat from Yuki. Have you classified the zooplankton groups according to their diel vertical migration behavior and connected this to estimates of their contribution to active carbon transport?
Yeah, that's a really interesting topic. So Bruce Robeson had a paper recently where he did look at specifically the vertical migrators and partly how that encouraged interaction. Like some of the predators will just sit and they won't migrate, but they will take advantage of vertical migration passing through their feeding webs. So I would advise you to look to this paper by Bruce Robeson. I think it's called Running the Gauntlet or something like that. But, and that's obviously a really important part of the whole carbon transport is not just that they're making sinking particles, but they're swimming up, eating something, swimming down with that material. So yeah, we haven't done it so much with vertical migration. We don't know that for all the organisms, but we have tried to look at, instead of classifying them taxonomically, grouping them trophically, like functional groups and functional diversity. But I think that is a really potentially fruitful area of study.
Great, thanks. Will, you have your hand raised if you want to unmute.
Terrific stuff. I might have missed the very beginning, so sorry if this has already been covered, but the question is about plastics and their potential impact on the stories you were telling. Are they providing a surface for growth that's changing our ocean ecosystem in certain ways?
Yeah, again, I would completely defer to Anela on that question. She's got some publications about that and has actually done some ROV-based studies. So you could either look up or just ask her about that directly. But yeah, I would defer to Anela on that. Definitely, it's definitely there.
Great, thank you. I think there's a couple more questions for you in the chat, Stephen, if you
Okay.
don't mind looking at those as we switch over to our second set of exciting lightning talks.
My name is Teja Bakam. I'm a research fellow at the Smithsonian Natural History Museum in DC. I want to briefly comment on the importance of subsurface methane transport in terms of marine and geological carbon budget. So to give a context of marine methane blending, so on the left, what you see is most of the adductive fluxes. So marine methane fluxes are quite common in the continental margins. On the left, we see what we typically call as cold seeps. So these are advective fluxes. They are getting towards the water column. From the methane has been transported from sediment towards the water column, or close enough to the seafloor. And on the right, you see a global distribution of diffusive fluxes. So these are methane fluxes that are coming in, but getting consumed within the shallow sediment itself, so not really getting into the water column. But the general gist of the slide is that subsurface methane transport is quite common in the continental margin. We don't know yet much about the how much they are pervasive in the deeper ocean setting. There are evidences that they are quite active in the ocean trenches, for example. So that might be something in the future we might learn more about. And they are present regardless of the plate tectonic setting, active or passive margin. They're pretty widespread. In the overall, the methane flux, even though they are pretty common in the continental margin, not much of the marine methane is entering into the atmosphere, which is good from a climate concern. And the reason behind is that they are primarily consumed in the shallow sediments by microbial consortia that perform anaerobic oxidation of methane, and whatever else that gets still flux out onto the water column still gets consumed by aerobic methane oxidizers, and a very limited methane is entering into the atmosphere. So marine sediments is one of the probably the largest marine methane reservoir, but still is only because of this active microbial filtration, very less methane is interacting directly with the atmosphere or climate from an ocean perspective. But the other side of it is that even though the methane is being consumed by the microbes, there's an inorganic and organic carbon that's been generated. So the methane carbon is essentially being converted into organic or inorganic methane. And that is what was one of my interests in terms of methane carbon budget. And one of the way we wanted to understand how relevant is this methane carbon fluxes is that we tried to track down, this was one of my PhD project actually, we tried to track down all the possible pathways methane has been transported, and then try to evaluate that. The picture on the right is essentially the major marine / geological carbon budget fluxes. Where will they fit in? Is it any relevant of that regard? So when we looked at it, essentially, without going into the details of the pathways, where this is more of a detailed picture of how carbon flows within that anaerobic microbial oxidation consortium. So essentially what's happening is there's a sulfate and methane coupling that's happening in the shallow sediments at the sulfate methane transition zone, which is the SMTZ. But the main takeaway just is that even though the methane is being consumed, a large amount of dissolved inorganic carbonate is produced, and also a significant amount of carbonate precipitation happens in the system. And what we can see is that when we actually try to we did a literature survey of what is the conversion rates of how much DIC is produced from different processes and how much the inorganic part, the organic conversion is very less, but the inorganic conversion is quite high enough. So if you think about it, the average, at least from our estimate, we could come up with about close to 10 teramole of carbon has been fluxed outwards to the water column from this just a diffusive flux setting, and about 4 teramole of carbon is precipitated as autogenic carbonate. And if you think about it, that outflux is about greater than 20% of the entire global riverine DIC flux to the ocean, and the autogenic carbonates is comparable to about 15% of the carbonate accumulation that we know in the continental shelf. So when we put it in a global picture, that does become a significant enough of methane-derived carbon cycling. So we could actually put an answer that if we actually have to consider the methane-derived carbon fluxes in the overall geological carbon or marine carbon budget, we do have to incorporate the dissolved inorganic benthic fluxes to the water column. And I think I missed, and the arrow that is pointing downward is the autogenic carbonate precipitation. So those two factors does need to come in to play from a methane carbon perspective. And I'll quickly skim through this one to keep it on time. But we actually did look into a little more details about how pervasive is the autogenic carbonate. And we could actually, so a couple of points I wanted to emphasize is that if you purely think of how much autogenic carbonate has been buried in the modern oceans, it is the third largest carbon burial mechanism in marine sediment. And the picture here is kind of an estimated, based on what pore water data we have, we kind of try to speculate where or we find these autogenic carbonates. They might not be like large crusts, they are essentially nodules or tiny little precipitates within the sediment setting. And the second part is still a work in progress. So the inorganic carbon that is fluxing from the sediment towards the water column in these sites, what is it actually doing? That is a large enough, about 10 teramoles of carbon per year, right? So is it coming out as CO2 or is it coming as alkalinity? If it is coming in as alkalinity, that could be a buffer for the surface water acidification, for example, or can we actually do some evaluate in terms of their pH impact? That has been a work in progress. So Hongmin Yao, a collaborator of mine, and I, we've been trying to look through what we have, the data out there. We've been trying to compile the bottom water TA to DIC data and then try to match with the non-methane flux sites. It's been quite hard, but this is, I also want to put a plug in if anybody is interested in this kind of analysis. If you have done measurements of inorganic carbon and methane flux sites, I'd be happy to connect and build on this data that we've been building. So I don't have an answer to this question yet, but it is actually quite interesting to see how much of this methane-derived carbon fluxes is contributing in terms of ocean carbonate chemistry. So with that, I'll wrap up. So the summary is that we have a generally good global, I mean, it's still primitive, I would say, because a lot of it is an extension of the regional estimate. So we do know that even though methane has been consumed directly, not much of methane is entering or interacting directly with the climate from the ocean, but the methane-derived carbon cycling is indeed playing a much bigger role than we would have thought previously. And we need to include that, especially in terms of, for example, geological carbon budgets. In the past anoxic events, these processes would have actually much more larger in scale. And lastly, again, putting out that note that we still don't know really about the alkalinity to DIC ratio of this methane-derived DIC outflux to the bottom waters, and something that might really help in overall ocean carbon chemistry in general. And with that, I'll wrap up. I want to give a shout out to my collaborators.
Thank you.
And also all the data sets that are out there. Thanks.
Great. Thank you so much. Okay, now we can hear from our next speaker.
Hello, good afternoon, everyone. My name is Shen Mao. I am currently a postdoc at Oxford Oceanography Institution. My lightning topic today is carbon export and flux attenuation near Bermuda. July 2016 case study of 1D biogeochemical model. Thank Heather King and Amy Mass for providing guidance on this project. So as for this collaborative research project, we focus on reconciling different carbon export and flux attenuation pathways at beds. So there are three different export pathways, including the particle thinking, mixing of dissolved organic carbon, and active export by zooplankton diurnal vertical migration. So, however, we still lack a very clear understanding on how this active PUC and DUC transport driven by DVM contributed these additional unaccounted sources of organic carbon from the euthic zone to the mesopelagic zone. So firstly, observational data is a key to understand these processes. By collecting the zooplankton biomass and abundance at different depths in July 2016, we can obtain the day minus night biomass in the left figure and the day minus night CO2 in the right figure from different size classes of zooplankton. And we can infer that a different size class of the zooplankton has contributed to this varying level of active CO2 flux at different ocean depths throughout the water column. And the first step from the observational data to modeling is to build this full size particle size distribution PSD functions between the active carbon flux and equivalent spherical diameter ESD. And this plot shows the different PSD functions at about 150 meter, 300 meter, 500 meter, and 800 meter depths. And we are trying to take advantage of all the available observational data collected by the AMI. And the ESD here is a metric used to describe the size class of the zooplankton. And we can infer from these four plots that it's obvious that magnitude and slope and even the sign varies significantly at different ocean depths of the water column. And then we apply those PSD functions into our original model structure. So we extend our model depth to the 1,000 meter depth with 25 meter vertical resolution. As you can see in the left figure, in the model, we have three phytoplankton groups, heterotrophic bacteria, protozoan, metazoan, nutrient, dissolved organic matter, particulate organic matter in this batch 1D of our model. And this 1D data assimilative model that assimilates batch observations has focused on evaluating the role of the heterotrophic bacteria in controlling the net community production, where they are found to be the dominant driver of the community respiration. But after implementing the PSD function into this model, we're able to investigate the DVM effects on different carbon export. This plot shows the temporal and vertical distribution of different form of the organic carbon under the baseline condition without the DVM. And the black line here represents the mixer depths. Labile DOC concentration are highest in the upper ocean, particularly within the mixer depths. And the semi-labile DOC concentrations are more broadly distributed throughout the water column. And PUC, we can see that it's concentrated in the upper ocean, but shows a very strong seasonal variabilities. So, so this slide really highlights the active PUC flux induced by the DVM. And this anomaly is defined as DVM case minus control run case in July 2016. And this plot shows the temporal and vertical distribution of PUC anomaly in July 2016. Okay, so due to the DVM effect, we can find in the upper ocean, zooplankton consume more phytoplankton and other particulate matter, reducing the PUC concentration in the upper ocean. As for in the mesopelagic zone, the zooplankton residing at the depth excrete fecal pellets and experience mortality, both contributing to increasing PUC levels in this mesopelagic zone. And this active transport mechanism really enhances the carbon sequestration in these deeper oceans. So this slide just summarizes and wrap up the take-home message. So this model successfully incorporated the effect of zooplankton DVM scheme, and at beds, the PUC concentration can be around 0.12 millimole carbon per cubic meter, and the calculated transport of 1 to 10 to minus 2 millimole carbon per cubic meter from upper ocean to the deep ocean represents a small fraction of the existing PUC concentration. However, over the extended period, its daily increments can contribute to more substantial range change in PUC, influence the carbon cycling at beds. I think I will stop here. Thank you.
Thank you very much.
Thank you.
Great job. So now we will hear from our next speaker. Take it away, Kayla.
Hi, thanks. Today I'm going to be kind of introducing and sharing some of the preliminary results from my new inverse model of the steady state biological carbon pump. So we're focused on parameters that are associated with subsurface processes, the ones that are uncertain, like getting like transfer efficiency and carbon fluxes to deeper ocean layers. I'm advised by Francois Primo. I'm at UC Irvine. So what I'm calling it right now is OxCarb because we're using oxygen to infer carbon export and remineralization. OxCarb is a really relatively simple single tracer model that relies on it's optimized with World Ocean Atlas and GLODAP data. So this is showing the oxygen concentrations from World Ocean Atlas on the left and then from OxCar on the right. And at deeper depths, oxygen is decreasing, and it also the match is pretty good, and it's capturing that OMZ in the equatorial Pacific. The steady state oxygen equation, because this model is a steady state model, I'm trying to get it constraining the baseline biological carbon pump, like parameters related to the BCP. The equation is setting the change in oxygen with time. So in a steady state model that goes to 0, plus the like physical mixing and transport, advection and diffusion, which comes from OCIM, the ocean circulation inverse model, equal to oxygen sources minus oxygen sinks. There's a lot here. I have a QR code at the end so that you can see these slides and more, but with the equation that goes into the model, what I want to draw your attention to is the respiration term, because that's like what's actually unique about this compared to other like OCIM-based steady state models. It's the only part of this model that has adjustable parameters, and we're basically saying that we're assuming that the vertical flux of organic carbon is going to decrease according to a power law with depth below a reference depth, so about 100 meters in the model. So this looks similar to the Martin curve, where our adjustable parameters in the most simple configuration in this model are A and B, so like a scaling coefficient, and A and then B being how rapidly oxygen is consumed with depth. We use the stoichiometric ratio of oxygen to carbon to transform oxygen utilization into carbon remineralization, and then can integrate the remineralization rates to get like export flux at every depth. Thank you. So this is showing like a sweep across the two adjustable parameters A and B. Like as a function of those parameters, the plot's showing the model data mismatch. So the more pink, the more red is saying that there's more mismatch between the oxygen produced from this model versus GLODAP and World Ocean Atlas. And the black contours are showing total carbon export. So the export at 100 meters, so the base of our reference depth euphotic zone. And then because A is kind of related to the total production at the surface, and B is kind of related to transfer efficiency, that's MPP that's estimated from the models on the y-axis, and then transfer efficiency, which is the ratio of the flux of carbon at 100 meters to oh, that's backwards. It should be the ratio of 1,000 meters to 100 meters. It's like a metric of efficiency for the BCP. That's on the x-axis. So you can see that white stripe is kind of like the valley of good models. The very most optimal that we get in the middle of that banana valley, whatever you want to call it, is making our estimate of total flux at the base of the euphotic zone about 11.5 petagrams of carbon a year, which is like cool. It supports, it's consistent with other OCIM-based approaches. And then it's like within the range of, I was a data analyst for the biological pump chapter of the Recap 2 report that came out last year. And so it's within that range, and it's also tighter than the range that we see that's estimated from prognostic models, which were like 5 to 13 petagrams of carbon a year. So it's like simple but powerful because it's just this oxygen mass balance approach. And it doesn't rely on a lot of assumptions. The next steps is that OSIM, the physical circulation model, the steady state one, there's a new version, cyclosim, that includes 12 seasonal time steps. So I'm going to incorporate this framework now that I'm seeing that it is kind of working to cyclosim to get the same results for that. And then also evaluate the sensitivity of mostly temperature and cell size. I think we're going to use the sensitivity of those in remineralization, like use those to scale A and B. And then also you could use this exact same framework with phosphate, which I know that other labs, I think presenting here are even doing. I want to repeat that, compare it, and then also do it with stable carbon isotopes because, they have a similar vertical structure, like because of the imprint of remineralization. First lightning talk, my timer didn't go off, so I think I'm within the time range, but I'm gonna end there. Yeah, you can scan the code if you want to look at the more more slides and see these. But yeah, these are preliminary results, and I'm excited to share it with you guys, and that's all.
Great. Thanks, Kayla. That was awesome.
Thank you.
For time, we should move on to our next speaker.
Okay, yeah. Hello, everyone. I'm Randie Bundy from UC Santa Barbara. I'm also working on data simulated model, and today I want to introduce our new findings in transfer efficiency with our latest seasonal data simulated model. Thinking particulate organic carbon as a dominant biological pump pathway attenuates with steps due to microbial degradation or zooplankton feeding. The efficiency of this pathway can be measured as the exported fraction, the fraction of exported POC to reach given depths in the mesopelagic or basipelagic zone, which is called transfer efficiency. And this metric can, in another matrix, as the POC flux is often described with a power law function of depth. The power law exponent B can be used to represent the attenuation rate. The lower B value is equivalent to a higher transfer efficiency. Over the decades of research, there is still no consensus on the latitudinal variations in transfer efficiency or B value. For example, a global synthesis of deep sediment trap data at the thorium-derived carbon export found transfer efficiency is lower at high latitudes, but higher at low latitudes. In contrast, the observations with neutrally buoyant sediment trap data found the transfer efficiency is higher at high latitude, but lower at low latitude. These observations are very sparse and biased towards summer season. Besides, the previous data constrained models can only simulate the annual mean state of POC flux and overlook its seasonality. Now we have developed a new data assimilation model to resolve the seasonal cycles of biological pump. The model is driven by satellite-derived NPP product. The chemical traces are transported throughout the ocean with the new seasonal varying circulation model called cyclone. And all the model equations are now solved in a seasonal steady state, and the model results are constrained by the multi-decadal observation data. Here is my model results. The spatial pattern of the annual mean transfer efficiency is similar to previous model results. In general, the transfer efficiency is higher in the subpolar region, followed by equatorial region, and it is lowest in the subtropical gyres. The model also resolves various seasonal variability in different ocean biomes. In particular, in North Atlantic and North Pacific, the transfer efficiency is lowest in early spring and higher in summer, while the transfer efficiency in Southern Ocean is lowest in summer but highest in winter. We found this model results can reconcile previous contradictory latitudinal variations from observation data. In the left figure, the asterisk represents the observation value, and the dot represents the model results sampled from the same sampled location and season. And you can see our model results match the observation value generally well. In Hansen's observation, his data, her data concentrated in the Southern Ocean and equatorial region. In the summer, the Southern Ocean, the transfer efficiency in the Southern Ocean can be as low as subtropical region. In masses sampling, his data focuses on Northern Hemisphere. In summer, the transfer efficiency in subpolar North Atlantic and Northwest Pacific is much higher than the subtropical region. So here is another question: why previous model study cannot find the north-south asymmetry? So compared with previous annual model, we found they overestimated the transfer efficiency in the upwelling Antarctic zone. That is because the annual mean circulation model OCEAN uses the winter mixed layer depths. The Southern Ocean is dominated by small phytoplankton, and the small particles are more susceptible to winter mixing. In the right panel, I take the POC profiles at the station SOTS as an example. You can see the vertical profile from the annual model is close to the summer profile in our seasonal model. So in summary, our seasonal data simulated model found transfer efficiency has opposing seasonality in subpolar Northern Hemisphere and the Southern Hemisphere. We found the seasonal sampling bias can interpret the contradictory latitudinal variations from observation. We also found previous annual models could overestimate the transfer efficiency in the Southern Ocean due to the usage of the winter mixed layer depths. And the preprint version of our manuscript on the seasonal carbon export and sequestration will be available online soon. And we have also uploaded our model output on Zenodo, and you are welcome to use our model product in your own research. That's all. Thank you.
Thank you very much. Great job, all of our lightning talk speakers. We really appreciate you speaking in our session.